point. I hope you guys enjoyed your extra hour of sleep. As I mentioned, today is going to be the last day where we have to talk muscle physiology. As I hinted at in the last class, basically what that means is first telling you that everything we've talked about so far about skeletal muscle is a big bold lie. And then we will spend the rest of the class time briefly talking about how smooth muscle and cardiac muscle are similar in their function to skeletal, but are not quite the same. And so we'll see those big differences. Those are things that will be emphasized more in 431, which is why we're not spending as much time on it, especially with a Wednesday exam. We also need to finish up our origins, our insertions, and our actions today. But as we saw, we got pretty far. So I don't see any reason why we shouldn't have a little time at the end of class today for an exam review. Again, remember, reviews are not me telling you what I think is important, but the opportunity for you to ask questions about the material we covered so that you can be successful on the exam, which is going to be Wednesday, the 10th. Both lab and lecture exams, you know what to expect from that. You've taken 3 of them already. And this is no different than the previous ones that you've taken, so you know what to expect with this. However, it does look like this is our first Wednesday exam. As such, we have 5 days before the next class. And while I want you to have a little bit of a break, take a good 30, 35 minutes after the exam to rest and recover, but to encourage you to look ahead at the nervous system on Monday, November 15th, you have two, how you spell Monday? 2 pre-labs. And again, the pre-labs are not the reviews. They are pre-labs. If you think about our lab section, there is the activities that we could be doing. There's the review at the end, but at the very beginning of our units is a pre-lab. So the pre-lab for unit 12 is going to be due, and that is pages 315 through 316. And the pre-lab, again, not review, for unit 13 is also going to be due. So that'll keep you busy a little bit over the weekend. And that is pages 334 through 338. And that is it. All right, questions on any of that? I've been talking for 5 minutes now. I'm going to assume you guys can hear me. Nobody said otherwise.
Yeah, we got you.
Okay, excellent. Someone had a question?
Yeah, there was like a survey that I saw last night come up on Canvas. Was that like a graded assignment or just random?
It's neither random nor a graded assignment, as the same way that I monitor your learning process to see how you are doing in the classroom. Occasionally, and even instructors get graded as well. So this is your opportunity to comment on what you think I'm doing successfully in this class and what you think could be improved in this class. So I happen to be being evaluated this semester as well. So part of that evaluation is we've had instructors come in and sit in on our lectures. I don't know if anybody's been paying attention and noticed that, but also there's the opportunity for student feedback on that as well. So again, I should say it on there. I do get those comments, but there are 2 important things that I want to point out. First is that they're anonymous, so nobody's name is associated with them. And secondly, I do not get them till after the grades are submitted. So again, I would appreciate you taking the time to do that, to be honest in your assessment of me and the class, and doing that, and be fully comfortable and confident in that it is anonymous and that I won't see it till after. So again, help me to improve as an instructor by please being honest and taking the time. I know the fun part is to like grade, A's, B's, C's, whatever it is, but the comments I think are the things that really help from instructors to be successful moving forward. So I would appreciate if you took the time to do that. So yes, thank you. I didn't know when that was posted, because again, that's something that is, I'm not controlling. The actually the district is actually who puts that on there. And like I said, I do not see that stuff till afterwards.
Okay.
Yep, great question. Any others?
And you're also doing on campus class next year for 432, right? But I saw that on there.
So yes, my understanding, and again, honestly, with 2 months, this still could change, but my understanding for next semester is that I will be having classes. My classes should be or are scheduled to be fully on campus. While there is been a strong push by many of the instructors in 430 and 431 to encourage the opportunity to still be online, both for the instructors who are more comfortable with it and for the students who it works better for, because I know all the questionnaires and evaluations they've done show about 1/3 of the class want to be on campus, 1/3 of the class want to be online, 1/3 of the class either don't care or want some kind of hybrid or something like that. So it is a very wide range of desires. The problem that we as a department are running up against is that now that California and Sacramento in particular is no longer in an emergency status, more and more of the nursing programs and even the dental programs are saying that now they're not willing to accept labs that are 100% online. They want hands-on labs, or they're not going to accept the classes. And so that's been the big problem we've been having, is trying to come up with some way of being able to provide that opportunity for students who are still interested in being online, but still making sure that the class meets the requirements, because it doesn't do you any good to take the class online if your nursing program isn't going to accept it. And so as of right now, with the condition of the way things are, my class is expected to be online. So the good news is it will be at the same time. So my 431 class, one of the things we have been trying to do even before COVID hit, is if you taught a class at a particular time, like 430 at a particular time, one semester, the next semester you teach 431 at that same time, because usually that's a time that works for students, you know, from one semester to the next. So I do have a 431 class. It will be from 8 to 12:30, just like this class is on Mondays and Wednesdays. The only difference is that as of right now, it is scheduled to be 100% online. I mean, 100% on campus.
Thank you.
Yeah, but again, who knows, in the next 2 months that could change. But as of right now, that is the game plan. All righty. Great questions this morning. Any others? All right, spectacular. Then let's dive in where we left off. We had learned everything you thought you ever wanted to know about skeletal muscle fibers, except for one big major thing. Skeletal muscle fibers is not a single thing. The same way that cartilage is not a single thing. We have talked about cartilage tissue and the characteristics of it. We know it's avascular. It doesn't regenerate very well. It has chondrocytes that live in lacunas. But we also know that cartilage comes in 3 flavors: hyaline cartilage, elastic cartilage, and fibrocartilage. Well, it turns out the same thing is true for skeletal muscle. Skeletal muscle isn't just one thing. There are actually 3 main types of skeletal muscle fibers. And those 3 main types of skeletal muscle fibers differ in some important ways. One way these skeletal muscle fibers differ is in the diameter of their cells. Now, we know that skeletal muscle cells are all cylinder shaped, but some have a larger diameter than others. Now, notice in parentheses there, when we talk about the diameter of the fiber, how wide the fiber is or thick the fiber is, it also says strength of contraction. Why would these 2 things be related to each other? Why would the diameter of a muscle cell affect the strength of its contraction?
Because there's more myosin heads for them.
Exactly. Remember, a thicker muscle cell has more proteins in it. And more proteins mean more myosin and actin. And more myosin and actin means more power strokes. So the thicker the muscle, the more proteins inside of it, and the more power, the more force it's going to be able to generate. Excellent. Another way that they can differ is in the type of myosin that they have in them. Remember, we mentioned how that myosin head performs a power stroke on an average of 5 times a second. However, with averages, that means some are actually faster and some are actually slower. And that is the case. There's actually 2 main types of myosin. Some myosin heads that are much, much faster, and some myosin heads that are much slower. And so obviously the speed of your myosin head determines the speed of your contraction. If you have fast myosin heads, what's the speed of your contraction?
Fast.
There you go.
Fast.
See how that, whereas slow myosin heads produce a slower contraction. Exactly. Remind me again what myoglobin is.
Stores oxygen.
Right, it stores oxygen, absolutely. And we talked about how it's an important inclusion in muscle cells and skeletal muscle cells in particular. But it turns out not all skeletal muscle cells have the same amount of myoglobin. Some have a whole lot of myoglobin, some have a little bit. And as we mentioned, myoglobin, like hemoglobin, stores oxygen. Like hemoglobin, it stores it with an iron. And so like hemoglobin in our red blood cells, when a muscle cell stores oxygen on its myoglobin, it gives it a more reddish color. Now, I know we don't usually think of muscle having different colors to it, but just a couple weeks from now is going to be Thanksgiving. And for some of us, this will be the first time in a couple years we've had a chance to get together with our family. And of course, what is one of the most important questions you get asked at Thanksgiving? I mean, there's all the really important stuff like, exactly. Like, who did you vote for and did you get vaxxed or not? And, when are you going to get a job? When are you going to have a family? When are you going to have more kids or some kids or all those kind of things? But as Sarah pointed out, the most important question is dark meat or white meat, right? What do you like, dark meat or white meat? Well, the dark meat is the muscle cells that have more myoglobin in it. So they tend to have more red fibers, giving them a darker color. Whereas the white meat has less myoglobin, and so it has less coloration to it. It is more whitish in color. Something you don't see on the on the dinner table on Thanksgiving, but is also important is capillary supply. Not all skeletal muscle cells have the same blood supply to them. Some of them have a large capillary supply, and some have a more limited blood supply. Why might blood supply be important?
Because it determines the amount of oxygen.
Yeah, because as we know, blood supply plays an important role in delivering oxygen to the muscle. So those that have a large blood supply are going to be able to get more oxygen. Those that have a limited blood supply are going to be able to not get as much oxygen. Right. Muscle cells differ in the amount of mitochondria. What are mitochondria used for again?
To produce ATP.
Yeah, exactly. Augustina's right. Absolutely. We use oxygen to fully break down sugar or glucose to make our ATP very super inefficiently. Right, so notice some are going to have a lot of mitochondria, some are only going to have a little bit of mitochondria. So notice with these, between the myoglobin, the capillary supply, and the mitochondria, some muscle cells are going to have a huge storage of oxygen, a huge supply of oxygen, and lots of mitochondria. So it's going to be very easy for them to break down the sugar with oxygen to make ATP, right, aerobically. Whereas others might not store as much, might not have as good of a capillary supply. So they're not getting a lot of oxygen. And if they're not getting a lot of oxygen, they don't need a lot of mitochondria. Notice all of these things relate to how the muscle cells are going to produce ATP. And when we talk about ATP production, as we mentioned, there's two ways to do it, with oxygen or without oxygen, aerobically or anaerobically, or, as we know, because anatomists hate us, there is a third way we can refer to these. Those muscle cells that use oxygen as their primary way of making ATP are known as oxidative, and those that do not rely on oxygen as much are known as glycolytic. So with these thoughts in mind, we can switch to our whiteboard and we can identify our three primary types of skeletal muscle cells. The first are fast glycolytic. There is a lot in the name. When we call them fast, what do you think that means?
They're fast.
They're bigger. Is the
Well, hold on.
strongest contraction.
Let's just talk about the term fast. Absolutely. When they're fast, as we mentioned, these produce a fast contraction. And why would they produce a faster contraction?
Because the myosin heads are moving faster.
Yeah, they contain fast myosin heads. Now, as someone mentioned, well, actually, let's do this. Move this down. Fast glycolytic are the most common type. That are found in the body. Like I said, they're fast. They produce faster contraction because they have fast myosin head. As I heard someone also say, these are the largest in diameter. What does that tell us?
It's the strongest contraction.
Yeah, exactly. This is the one that's going to produce the strongest contraction. So notice with that fast myosin head and that large diameter, these produce very fast, powerful contractions. However, as I just finished saying, there is a lot in a name. These are glycolytic. What does that mean?
Anaerobic.
Excellent. This means they primarily produce ATP. Was that with or without oxygen?
Without.
Without oxygen. Absolutely. So these, if they're not using oxygen to make their ATP, then that means they have a small amount of myoglobin. What would that do to their color?
Make them more like white.
Yeah, these are the white fibers. These have a limited blood supply. Right. These have a few mitochondria. Right. So when we talk about their ability to make ATP, they can make ATP really fast, but are these able to produce contractions that are going to be able to last for a long period of time?
No.
No, remember, this is that Hummer versus that hybrid, where basically one glucose is only giving us 2 sugars as a result of it. So these are muscles that will also fatigue rapidly. So these are muscles that can produce fast, powerful contractions, throw that pitch, throw that punch, make that jump, sprint. All things that are fast, that are powerful, but cannot be sustained for a prolonged period of time. Compare this to our second type. It's slow oxidative. Again, a lot in a name. Slow, what does that mean?
Low myosin.
Yep, exactly. These are going to produce a slower contraction. And the reason is because they contain slow myosin heads. Excellent. These are the smallest in diameter. What does that tell us?
Weaker contractions.
Excellent. These are going to be the weakest contractions. So notice these produce a slower, weaker contraction. However, there's a lot in the name. These are oxidative. What does that mean?
Use oxygen.
Yeah.
Aerobic.
So these are going to primarily produce ATP with oxygen. Now, of course, if you're going to produce ATP with oxygen, you need a large amount of myoglobin. What is that going to do to the color of our cell?
Red fibers.
Yeah, these are the red fibers. They're going to have a large blood supply. And they're going to have many mitochondria. So while these may not be as fast or as powerful, these are fatigue resistant. So we can use them to produce contractions that can last for a long period of time. As we said before, while you may not be able to sprint for hours, how long can you stand for?
A long time.
Yeah, you can stand for hours. Everything is walking distance if you have the time, right? So, and again, think back to that Thanksgiving turkey we were just talking about. Where's the white meat?
And breast.
The breast and the wings, absolutely. Where's the dark meat?
In the chest.
Thighs.
Thighs and legs are the dark meat, absolutely. So if you think about the turkey's life, how does the turkey primarily get around?
Walking.
It stands and walks. Is a turkey capable of flight?
Yeah.
Yeah, it can fly, but brief periods. They fly up to the top of the tree, or they fly over the fence. Or if you live here where I live, over by Mydew Park, there are a ton of turkeys. They'll fly over your car sometimes, right? So they'll be able to take flight for brief periods of time, but it's not their primary way of getting around. How many people here have eaten duck before?
Yeah.
Yeah, a couple of you. Excellent. Now, especially when you eat the breast of the duck, what does that look like?
It's dark.
Yeah, it's a much more dark meat. Absolutely. Because how do ducks get around?
They fly.
Flying.
They fly, right? Can they walk for brief periods of time? Or when they're sitting in the pond, can they paddle for brief periods of time? Yeah, but their primary way of getting around is flight. So their red meat is up here in their breasts. So what are we? Are we turkeys or are we ducks?
Turkeys.
We're turkeys, absolutely, right? We can throw that powerful punch or throw that powerful ball. But as we've talked about before, how long can you stand for?
Hours.
Ever. How long can you hold yourself in a planking position for?
A minute.
Minute.
Yeah, a couple minutes, exactly, absolutely. And remember, when you're planking, you're not even holding all of your weight. So we have much more of our fast glycolytic in the upper part of our body, more of the slow oxidative in the lower part of our body. Now, there is a third type, and again, that's all in a name. Fast oxidative. Let's start easy. What does fast mean?
A faster contraction
Why?
because it's got fast myosin heads.
There we go. Excellent. So let's see, if this is the largest and that is the smallest, then that means our fast oxidative must be intermediate in diameter, meaning that it has intermediate strength. Or actually immediate contractions. Say it that way. So again, this produces intermediate in contractions. Oh, let's say it this way. Contractions that are intermediate in speed and strength. Well, in strength, but they're fast. Excellent. And there's a lot in the name. What, of course, does oxidative mean?
Produces ATP with oxygen.
Yeah, although this is, again, one of those cases where our anatomists hate us. While fast oxidative have the name oxidative in their name, what's actually true for our fast oxidative fibers is that they are actually equally able to produce ATP, both with and without oxygen. So they're equally able to do it both ways. So that doesn't really sound like oxidative to me, but maybe they were just being lazy, didn't want to come up with another name, although they like to have lots of names for everything. So I don't have a good reason as to why. These are going to be intermediate in myoglobin. What is that going to say about their color? They're not white fibers, but they're not red fibers either. What does that make them?
Pink.
Yeah. These are pink fibers. Intermediate blood supply. And intermediate mitochondria. So while these can produce a fast contraction with an intermediate amount of strength, these are also going to be fatigue resistant. These are the ones that allow us to be able to jog for a prolonged period of time, right? Or swim laps in a pool. Not, I mean, like racing fast, but like leisurely swimming in a pool. Things along those lines. Now, every single muscle in your body, except for 6 of them, are a mix of all 3 of these fiber types. The 6 muscles in your body that are not are all involved in moving your eyeball through space. The muscles that allow you to move your eyes up or down or left or right or rotate them around, those muscles are all made up of fast glycolytic. And are those 6 muscles on your list?
No.
Nope. So you don't need to know them for this exam. For the next exam, you will get to learn them. The good news, it'll take you about 3 minutes. So that's actually, it'll take less than that. It'll take you about 30 seconds to learn those 6 muscles. So we'll save it for the next section of the class. But aside from those muscles that move your eyeball around in space, every single muscle in your body has a mix of all 3 types in it. Now, does every single muscle in your body have an equal mix of all 3 types?
No.
No, as we've talked about, we tend to have more fast glycolytic in the upper part of our body, more slow oxidative in the lower part of our body. But even that, in the bicep brachia, do we all have an equal amount of all 3 of these types?
No.
No. Remember, one of the things we talked about in the beginning of this section is if we wanted to give up on all of this and just work out for 4 hours a day instead of studying for 4 hours a day, we said we'd all get bigger. But as we also mentioned, if we all did the same amount of weight, the same number of reps for the same amount of time, would we all get the same amount of gain in size of our muscles?
No.
No.
No, and that's because we all have different amounts of the white fibers versus the red fibers versus the pink fibers in the muscles of our body. And what primarily determines that?
Genetics.
Yeah, genetics, right? And is it really possible to switch these? Not really, although there is a little bit, but it does get back to what the other concept we were talking about before. Remember, we were talking about those Olympic sprinters who have spent their entire careers focusing on maximizing the efficiency of their fast glycolytic fibers. So they get that powerful sprinting action that allows them to run 100 meters in less than 10 seconds. Whereas that marathon runner at the Olympics has been focusing on maximizing the efficiency of his pink fibers. Remember, we talked about the example of Usain Bolt, probably one of the greatest sprinters in the history of all mankind. Right? And he's world-class sprinter. And as we said, could he decide tomorrow that he wanted to be a marathon runner? He could
No.
decide, but would he be world-class tomorrow?
No.
No, because he's been maximizing the efficiency of all of these muscle cells. Now, with time and training, it is possible to convert some of these fast glycolytic to fast oxidative. So in time, it might be possible for him to retrain those muscles so that he could become a marathon runner, but that would take years and years to do. And again, he might not be able to convert enough of them where he would be able to compete at a world-class level. So genetics plays a huge role in determining how efficient these types of actions are going to be. All right. We've done it here on the whiteboard. Let's go back to the pretty words from the textbook. Again, you got a nice table that talks about this. The first most common type are the fast glycolytic. Remember, those are the large diameter white cells with low amount of myoglobin. Also few mitochondria, low blood supply. These are the ones that produce our ATP anaerobically. Right. Again, like that Hummer, if you own a Hummer, can you get away with having just a 10-gallon gas tank on a Hummer?
No.
No, you wouldn't be able to get from gas station to gas station if that was the case. So you have to have a huge fuel reserve. And the same thing is going to be true for these because they're so efficient. They produce that fast, powerful contraction, but they fatigue very easily, very rapidly. So like I said, these are the ones like lifting that weight, throwing that ball, throwing that punch, doing that leap. Rapid, intense movements that last for a very brief period of time. Conversely, those slow oxidative are the ones that let you wait tables for 8 hours a day. They are smaller in diameter, producing a weaker contraction, but they have a massive amount of myoglobin, store a large amount of oxygen, have a tremendous amount of mitochondria. They have a good blood supply, and they can produce their ATP aerobically. So while they produce a slower and a weaker contraction, that contraction can be maintained for a much longer period of time. All right, so this is what helps us to maintain our posture. This is what allows us to stand and to walk. And lastly, we have those fast oxidative. These are the in-between ones, in between in size, in between in myoglobin. And where they're not in between is how they make their ATP. They are equally able to make ATP aerobically and anaerobically. They have fast myosin heads that allow them to produce a fast contraction, but unlike the fast glycolytic, these are fatigue resistant. So these are the things that allow us to do that leisurely swimming, that jogging, those activities that can be sustained for a much longer period of time, but isn't just standing still. And again, like I said, not only is every muscle in your body, with the exception of the ones of your eyes, a mix of all three types, but even within the fascicles, there is a mix of all three types. Notice this great picture. Oh, you got your table there from the picture. Look at this great picture. This is a cross section through a skeletal muscle cell, through a skeletal muscle, where we can actually see the stain has been used here to stain myoglobin content. So notice this one here has a large amount of myoglobin, as does this one. These are our slow oxidative. Notice a lot of these have an intermediate amount of myoglobin. Those are our fast oxidative. And right here, smack dab in the middle, and two more up here, are some cells that have almost no myoglobin in it. So those are our fast glycolytic. Notice this is a pretty high magnification view. Here is a lower magnification view where we can see numerous fascicles. But again, we see this variation in color. Some have a large amount of myoglobin in them. Some have an intermediate amount of myoglobin in it. And some have almost no myoglobin in it. So notice with this particular stain that's stained specifically for myoglobin, we can see how much variation there is. And notice this particular muscle seems to have more of the slow oxidative than any of the other types in this. So this is probably for a human like a leg muscle or something like that. Maybe one of the hamstrings or something. Notice we can still sprint, fair amount of light, but a lot of dark. A good mix of all three types. All right, questions on that? All right, with that then, we are finally done with skeletal muscle. That is now truly everything you ever wanted to know and more about skeletal muscle. All right, so what we have left to do is smooth muscle and cardiac muscle. And even though I know it's a little bit early, this is a good natural break point. So let's go ahead and take our first break here. I will pause the recording at this point. I'm going to try to see if my interweb is back on and switch back to that, so I don't have to keep using my my phone for the hotspot. So if I get disconnected from the class, I will jump right back into it. But let's go ahead and take our break. We'll take a 15-minute break, come back at 8:55, and at 8:55, we'll pick up the lecture from there. Any questions? All righty, I will see you guys in 15 minutes. All righty. Any questions before we get started? So let's start easy. Tell me some things you know about smooth muscle tissue.
It lines hollow organs.
So most lines hollow organs, right? Remember, we know that there are some exceptions like the erector pili muscles. Walls of hollow organs. Uninucleated. Excellent.
Spindle shaped.
Spindle shaped. Excellent. What else?
Involuntary control by the autonomic nervous system and hormones.
By the autonomic nervous system and hormones. Excellent. I think you guys got most of it except for the easy one.
Highest regeneration rate.
Highest regeneration rate. There you go. The whole reason it's called smooth muscle. No striations. All right. So it is, as we now know, those striations are caused by sarcomeres lining up in the myofibrils. So if our smooth muscle cell doesn't have striations, then that means there's no myofibrils. And that also means that there's no sarcomeres. So if it has no myofibrils and no sarcomeres, can it use the same contraction method that we learned for skeletal muscle?
No.
No. Excellent. So absolutely, those are all great things that we know about our smooth muscle. And again, while this part here isn't necessarily new, the no sarcomeres, no myofibrils, we now understand if it has no striations, those things must be true. Excellent. But clearly it is muscle cell and it contracts, but it's going to contract with a different process. So let's take a look here. We see, again, notice many of the hollow organs actually have two layers, a circular layer, which would control the diameter, a longitudinal layer, which would control the length. So we see those differences. And notice we can see this is a cross-section view where we've cut the fibers this way, or if you cut the tissue so that you see this, this would be a longitudinal view. Excellent. As I mentioned, found in the walls of the hollow organs. And we'll go ahead and spoil the surprise to begin with. The same way there are three types of skeletal muscle, there are two types of smooth muscle. The most common type is the single unit, or what is also known as visceral smooth muscle. This is the most common type. And these are the ones that we were just talking about in the small intestine or in the stomach or in the blood vessels or any of those hollow organs. The advantage of them, as we mentioned, is that these cells line up in layers. And the reason they're called single unit is because there are gap junctions that connect these cells together. So that when my neuron comes in and forms a synapse and my autonomic nervous system stimulates this cell to produce an action potential, that action potential can spread to all the other cells, right? That's why it's called single unit, because basically these work together as a single unit. And in fact, some of this single unit smooth muscle, like for instance, in the digestive system, can be autorhythmic. What does autorhythmic mean again?
Contracts on its own.
True, absolutely. It contracts on its own because it can produce its own action potentials. You actually have this elaborate network, what's called the enteric plexus, which is a term you may not have heard before, but it is sometimes also referred to as the gut brain. When you had that nice big cheeseburger for breakfast and it reached your stomach, did you have to consciously think, all right, stomach, you have food inside of it, time for you to be able to start doing its work?
No.
No, what actually happened is when that cheeseburger hit your stomach stretched. That signal from the stretch of the stomach goes to this enteric plexus, this elaborate network of nerves in the stomach, where it makes a very basic decision. Hey, I'm being stretched out. There must be food inside of me. It's time for me to start churning that food and releasing digestive juices to break it down. And all of those decisions were made right there in the stomach. We didn't take any kind of conscious or voluntary or involuntary control from the brain. So some of it, not all of it, but some of it actually has the ability to be able to regulate its own reflexes. And like I said, the key to this is it's able to work together because it has those gap junctions. However, the erector pili muscles, for instance, on the other hand, are what are known as multi-unit. In this case, it is much more similar to skeletal muscle. Muscle cells are bundled together into a fascicle-like structure. And much like the skeletal muscle cells in a fascicle, each individual muscle cell requires its own stimulation so that it can be stimulated so that they can contract to make your hair stand up or change the diameter of the iris of your eyes or move your lens so you can focus up close or focus far away, things along those lines. And of course, your book's got a pretty picture emphasizing this point as well. I don't know why it had to make the multi-unit look so messy. I think it was just trying to emphasize that it doesn't arrange in layers, it arranges in bundles instead. All right, now where smooth muscle is really different is in its anatomy. So here's the pretty picture from your textbook. But let's, like we've done before, go ahead and draw this and simplify things a little bit. Make sure we understand the key components to it. I'm going to cheat because I know that's not quite a spindle shape, but this diamond shape will do a better job of emphasizing what we're doing with our smooth muscle cell. Now, as we know, it has a single nucleus located in the middle, but that's going to be in our way. So I'm going to go ahead and remove that from our picture. So let's talk about some of the big differences. Notice if we don't have myofibrils, we don't need a triad. In fact, there are no transverse tubules. Remember, transverse tubules were those big, huge invaginations of our plasma membrane, travel deep into the muscle cell. However, there are some small pockets on the surface. So let's use a color for this. There are small invaginations, these little pockets that can be found on the surface of the plasma membrane of this. These are what are known as the little caves or the caveolae. Not only is there no transverse tubules, but there's a very limited sarcoplasmic reticulum. So again, no triad. And with a limited sarcoplasmic reticulum, remind me again, not only is there no triad, there's no sarcoplasmic reticulum. I should have said it this way. Let's do this. Cheat and get rid of that. Not only is there a limited sarcoplasmic reticulum, but there are no terminal cisternae. So no triads. And why is the terminal cisternae and a large sarcoplasmic reticulum so important in skeletal muscle? What does the sarcoplasmic reticulum do in skeletal muscle? Yeah, store calcium, absolutely. So our smooth muscle does not store a lot of calcium. Instead, it relies much more on extracellular calcium. And that's actually what those caveolae do. The caveolae are little pockets on the outside of the plasma membrane that store calcium. Keep the calcium close so that when it needs it, it is able to come inside. Now, to contract, we still need contractile proteins. And what are the contractile proteins again?
Myosin and actin.
But there are going to be some slight differences in them. Our myosin is still a thick filament, and it still has myosin heads coming off of it. However, remember the myosin heads before pointed away from the M line. There is no M line on this. So what's interesting about our myosin in the smooth muscle is that all the myosin heads point one way on one side, and all the myosin heads point the other way on the other side. We still need actin to pull on. And if the actin is going to have some effect on the cell, I know my drawing here isn't the best, but we'll get to it. It needs to be anchored to something. Normally it is anchored to the Z discs, but in this case, it is anchored to a structure that is known as the dense body. These dense bodies are actually connected to the sarcolemma. So they're connected to the plasma membrane, and they have cytoskeletal structures are intermediate filaments that connect to them. So there's going to be oops. Forming kind of like this mesh-like structure around the entire muscle cell. So we have these dense bodies and these intermediate filaments interconnected, myosin and actin in between. So when our muscle contracts, as we know, myosin heads are going to grab and pull on the actin. And so they're going to pull this actin this way, and they're going to pull this actin this way. And so our dense bodies move towards each other. Kind of like what you see in our skeletal muscle cell, where our muscle cell, I'm going to make this smaller so I can fit this all in here. So pull on the actin, pull on the dense bodies, get them closer together, kind of like the Z discs get pulled closer together. But notice our dense bodies are interconnected. So when one dense body gets pulled, it yanks on the intermediate filaments of the ones that are next to it. And of course, if this one pulls, then it's going to pull on its neighbors, and so on and so forth. And then when this one gets pulled forward, it's going to pull on its neighbors, and so on and so forth. So notice in this case, all of the movement isn't in one direction. With our skeletal muscle, one of the reasons it's so powerful is everything is working on making it shorter. Here, it's getting shorter, but it's also twisting and bulging as it contracts. So as this contracts, like we expect, the muscle cell gets shorter, but it also twists and bulges. Now, with this kind of twisting and bulging motion, is this going to produce a very fast contraction?
No. No.
With all of the forces going in different directions, is this going to produce a very powerful contraction?
No.
No. So notice these produce these weak and slow contractions. In fact, this contraction can be as much as 30 times slower than the contraction in a skeletal muscle cell. However, because it's all interconnected, is it going to require as much ATP to be able to maintain this contraction?
No.
No. And so while this is much slower and much weaker, these contractions can be maintained for a much, much longer period of time. Oops, maintain. So we can maintain that muscle tone in the stomach, or we can produce powerful, slow contractions of the large intestine during defecation, right? This type of movement provides for a very efficient contraction. So it may be slow, it may be weak, but it can be sustained for a very long period of time. Now, I've done an okay job here drawing this, but let's look at the pretty pictures from your textbook. Again, notice we have to have our thick and thin filaments. We must have our myosin and our actin. But they're not arranged in sarcomeres. No transverse tubules, very little sarcoplasmic reticulum. We do have those caveolae that are the little caves that contain the calcium on the outside of the plasma membrane. And inside, we have our dense bodies anchored to the plasma membrane and the intermediate filaments that anchor them together, twisting around the entire smooth muscle cell. So, as the actin and myosin pull on those dense bodies, not only does our muscle cell get shorter, as we see here, but we get this twisting and bulging of the muscle cell as well. Produces a much, much slower contraction than we see in skeletal muscle. But again, this is a contraction that can be maintained for a much longer period of time. Right? The job of the stomach isn't to, pound and beat the hell out of that cheeseburger you had for breakfast. It is to produce these smooth, sustained contractions to churn and mush that food up. Right? I know every single person in this class, when they take a bite of food, they chew it the 30 times you're supposed to before swallowing. But some people will take a bite, chew twice, and down the gullet it goes. So for those people, having this twisting, churning motion of the stomach helps them in the mechanical breakdown of that food. And of course, I think we've used this term already in this class, but just as a reminder, the other thing that this single unit visceral smooth muscle can do, especially with two layers, as we mentioned, we have that circular layer and that longitudinal layer. So we squeeze the small intestine, then we shorten it, and then we squeeze it and we shorten it, and then we squeeze it and we shorten it. And as we do that, we're able to move the cheeseburger through our small intestine, and those rhythmic contractions of the smooth muscle to move substances through their hollow organs is called peristalsis. All right. Questions on that? All right, that is what we need to know about smooth muscle. Tell me, and I don't have a lot of room to write here, so let's cheat. I'll steal a chunk of space right here. Tell me what you know about cardiac muscle.
It's only found in the heart.
Excellent. Striated. And again, now we understand what that means. If it is striated, oh, these are awesome. Hold on, slow down. You guys are doing great. Striated, remember, now we know that means that it has myofibrils. It has sarcomeres. And so it is going to use a similar contraction mechanism to our skeletal muscle. So myosin is going to have that contractile cycle where the myosin is grabbing on the actin, pulling on the actin, pulling on the Z discs. Everything gets shorter, everything gets closer. Excellent. We know what that striated means. Let's see what else you guys had here. No regeneration. And also these have autorhythmic cells.
And involuntary.
Well, so right, this has autorhythmic cells, so they can produce their own action potentials, driving and limiting their own force. But you are correct. So we don't need our hormones and our autonomic nervous system to control them. Instead, we use our autonomic nervous system and our hormones to modify the rate of contraction. Remember, as we talked about, if I ripped your heart out of your chest, put it in a petri dish, and provided it with all the oxygen and the nutrients it would need, it would beat at a rate of about 100 beats per minute, because that's the rate it wants to produce its action potentials at. It wants to produce its contractions at. In your chest right now, unless you had a quad shot from Dutch Brothers, it's probably not beating 100 beats per minute. So instead, we're using our autonomic nervous system to modify. Excellent, branch shaped. Anything else?
They have intercalated discs, which means they have a lot of desmosomes and gap junctions.
Excellent. So cardiac muscle cells connect to other cardiac muscle cells with that intercalated disc. Oops, 1L. Intercalated discs, which, as you mentioned, which contain a large amount of gap junctions, which, as we now know, allows them to work together as one. And also a large amount of desmosomes, which, as we know, hold the cells together tightly. Excellent, so that they don't shear apart. Excellent. Most are uninucleated, but there are a few that can have two nuclei. Excellent. Spectacular. Can't think of anything else. You guys got anything else? I think those are the big stuff. All right. Excellent. Yes, Augustine, you had a question?
On a test, if we're asked if it's uninucleated or multinucleated, what would we say? Like, would it be like which one would be?
If those are the only two choices, uni and multi, then definitely say uni,
Okay.
because they're definitely not multi. Yes, there are a couple that might have two, but I definitely would not say, I mean, you look, even at the illustration here from the textbook, every single cell that they've got here has just a single nucleus in it. So again, your book mentions it and your histology mentions it. There are occasional cells that you would find that have two nuclei, but they're not common. So if you were to ask me to describe, cardiac muscle cell, I would say it's uninucleated.
Okay, thank you.
Yep. Excellent. Great question. Any others? All right. While it uses a similar mechanism for contraction, clearly there are some differences anatomically. As we mentioned, it has a single nucleus and it's located in the center, unlike our skeletal muscle cells where it gets pushed to the side. But notice there are some other differences that we see here as well. We have myofibrils. Our myofibrils are made up of sarcomeres that have Z discs, that have M lines, that have actin and myosin. So there's an H band and an I band and an A band and an M line and all of those kind of things. Notice we have transverse tubules, invaginations of the muscle cells to carry that action potential deep into the muscle cell. Invaginations of the plasma membrane, the sarcolemma. Mitochondria, lots of mitochondria because we need to be able to make our ATP aerobically. But notice it has a very scant sarcoplasmic reticulum and no real terminal cisternase. So notice there's no triad, no terminal cisternase. And so notice once again, because of that less extensive sarcoplasmic reticulum, this cell also relies more on extracellular calcium to move the regulatory proteins. Right? With our skeletal muscle, pretty much all the calcium we need to move the regulatory proteins is right there locked up inside the cell all the time. But that is not the case with smooth muscle, definitely, but it's also not the case here with cardiac muscle as well. It relies much, much more on extracellular calcium. So, similar anatomy, so not surprisingly, similar function. And the good news is this is stuff we've already learned. Let's go to our whiteboard again. And let's talk about some things we know. One of the things we know is that a skeletal muscle produces an action potential. Let's do this. Oops. Again, anytime we have a graph, we want to label it time down here, voltage up here. We know that we have a resting membrane potential. What is the resting membrane potential?
-70 millivolts.
Excellent. And we know we need to get this cell to produce an action potential. We need to get it to a threshold. And what is that threshold?
Sixty.
60 millivolts. Perfect. We'll do it that way. Excellent. So we know our skeletal muscle cell happily stays at its resting membrane potential till it's depolarized and reaches threshold. And when it reaches threshold, it produces a skeletal muscle action potential, which is a big positive signal. We get a rapid depolarization, but we also know this doesn't last for a long period of time. So it just as rapidly comes back down and goes at that resting membrane potential. So our skeletal muscle action potential lasts somewhere between, I don't know, 1 and 3 milliseconds. It's an incredibly fast process. However, as we also have learned, a muscle action potential, a single muscle action potential, isn't typically how I move my body through space. I need to be able to generate multiple action potentials. However, is there a limit to how fast I can stimulate the muscle cell to produce that action potential? Yes. Muscle cells have a brief period of time where after they're stimulated, they lose their excitability. And what was that period of time called?
Refractory period.
Yeah. It has a refractory period where we get that rapid depolarization, rapid repolarization, and it has a refractory period. And a refractory period, remember, is only about 1 to 5 milliseconds. So it is a very, very short period of time. So we have a rapid depolarization. Remember, this is a big positive signal, about a 100 millivolt change. And then that is followed immediately by rapid repolarization, where the cell goes back to rest. And of course, we know that one neural action potential produces one muscle action potential, which produces what?
On twitch.
There you go. So the other piece of information that is not new that we already understand is that we can have a skeletal muscle twitch. And again, this is something we can draw. So again, we'll put a graph. And anytime we have a graph, we have to label our axes. Time. I always make a point of emphasizing this because when we're in the classroom, I make you draw these. And you, of course, lose points if you don't label them. You don't have to draw graphs on this test because it's online and drawing is not easy. So you're getting off the hook, but me saying it is something that apparently is ingrained. So excellent. We have our tension, we have our time, and we know that that twitch has 3 phases. What are the 3 phases, of a twitch? Latent, excellent. And remind me again here, let's do this. Here is when we generated our muscle action potential. And after we generate our muscle action potential, how does our tension change during the latent phase?
There's no tension change.
There's no tension change. So there's a brief period of time where the tension remains 0. What does happen? Does that mean nothing happens during the latent phase?
Regulatory proteins are being moved.
Well, it's more than that. It's this, again, our muscle action potential is spreading down the axon, spreading down the transverse tubule, depolarizing the triad, which opens up the voltage-gated calcium channels, which allows calcium to come out to move the regulatory proteins. So if you think about it, during the latent period is when our excitation contraction coupling is occurring. So again, it isn't just about moving the regulatory proteins, it's about getting all of that stuff done so that the regulatory proteins can be moved. And so like we said, during the latent phase, there is no change in pressure, in tension. What is the second phase? Contractile phase. And how does the tension change during that?
It increases.
Excellent. So let's do that. Then what happens?
Relaxation.
And what happens to the tension during that time?
Decreases.
Excellent. And why are we seeing this increase and decrease in tension? What's causing it?
Calcium.
Yeah, there's more calcium available, more calcium coming out. So we get an increase. So increase in calcium means more regulatory proteins, mean more power strokes. So tension goes up. Relaxation, that calcium is put away. So as calcium levels go down, fewer regulatory proteins are moved, meaning fewer power strokes, meaning fewer, less tension, until it goes back down to 0. And on average, what did we say time-wise a single twitch would take?
40 to 100 milliseconds.
Yeah, 40 to around 100 milliseconds.
Do we have to spell out milliseconds on the exam?
No, you could just put MSEX, yeah. SEC, yeah. MSEX is something totally different. But yes, you can just abbreviate MSEX. MSEC. All right, excellent. All right. So notice there's no new information on the board right now. This is just emphasizing the things that we have already talked about. Well, it turns out the reason that we're doing this is because there are also cardiac contractile cells. Let's actually say it that way. And those cardiac contractile cells produce a cardiac muscle action potential. Once again, these cells have a resting membrane potential. Once again, these cells have a threshold. And they're the same that we saw in skeletal. Threshold equals negative 60 millivolts. The resting membrane potential equals negative 70 millivolts. And with these contractile cells, much like with our skeletal muscle, if you leave it alone, it will happily stay at its resting membrane potential forever. Until you stimulate it. When you stimulate it to threshold, just like with skeletal muscle, we get a very rapid depolarization. So we stimulate. And we get a massive depolarization. And as I think we talked about before, this is primarily from sodium entering the cell. So we have this massive influx of sodium that comes into the cell, just like over here. We have a huge influx of sodium. So far, they look the same. However, here is where our cardiac muscle starts to differ. Instead of immediately depolarizing, the muscle cell stays depolarized for a prolonged period of time. It has a prolonged depolarization known as the plateau stage. And the reason for this is threefold. Yes, our sodium stops coming in over here, but then very rapidly, sodium stops coming in. And also potassium leaves the cell. And so it's that potassium being a positive ion leaving the cell that makes the cell go back to its resting membrane potential. However, here it starts the same. Sodium stops entering. But potassium permeability decreases. So potassium, the positive ion, stays inside the cell. And remember, we have all that outside calcium coming in. And that calcium entering the cell and the potassium staying in the cell keeps the cell depolarized for a prolonged period of time. Eventually, our potassium leaves, and when our potassium leaves, we go back to rest. And when the potassium leaves the cell repolarizes. However, this action potential is much, much longer. This action potential can last for well over 100 milliseconds. It can be a much, much longer depolarization. Now, of course, when our cardiac muscle contractile cells, and guess what contractile cells do? Not a trick question. What do you think contractile cells do?
They contract.
They contract. There you go. So when they contract, they also produce a twitch. And just like we saw before, right? One muscle action potential is going to give us one twitch. And so when we look at this twitch and draw this twitch, it's going to have the same three phases. So when we generate our muscle action potential, we are going to get a latent period, followed by, actually, I'm going to cheat a little bit. Followed by a contractile period, followed by a relaxation period. However, if we have a prolonged muscle action potential, what do you think happens to our twitch?
twitch will be longer?
In fact, the twitch here can be around 200 milliseconds. Why? Why have a longer twitch here in our cardiac muscle cell? All right, let me ask a different question. How many of you have ever inflated an air mattress before?
Yeah.
Mm-hmm.
A couple of you, excellent. And I don't mean the way you do it now where you plug it in the wall and you push the button and it fills up. No, I mean out camping with that hand pump.
Yeah.
Okay, absolutely. Some of you have. Is it a fun experience?
No, it takes so
not
long.
particularly. However, while you're doing it, do you make a whole bunch of rapid, small, quick pumps like that? Or do you go the full length of that pump while you're using it?
The full length.
Why?
It's more efficient.
Yeah, that prolonged pump moves the air more efficiently. Well, your heart pumps blood. So do you think a bunch of fast, rapid contractions or a long, sustained contraction is going to be more efficient at moving the blood?
Long sustained.
Yeah, so this long, sustained contraction makes it a much more efficient pump. Now, again, one last thing. When I'm moving my body through space, usually I don't use just one twitch. I add twitches together, all right? I tetanize the muscle to get that nice prolonged contraction, sustained contraction. Do I want to necessarily tetanize my cardiac muscle where it locks up in a prolonged contraction? No. So the last thing we want to make sure we emphasize is we don't want to add waves together. We do not want this muscle cell to tetanize. And the way we do that is by having one more thing prolonged. We have a prolonged refractory period. The refractory period for our cardiac muscle cell is around 250 milliseconds. Notice by the time the cardiac muscle cell is ready to be stimulated a second time, it's already done contracting. It's already done back at rest. So it is impossible to add waves together. It's impossible to tetanize the muscle. And that's absolutely positively a good thing. All right, we've done it here with the pretty pictures. Let's go back to the lecture and emphasize these things. Again, our muscle action potential for our cardiac muscle lasts for a much, much longer period of time. We have that prolonged plateau period, primarily from the influx of calcium coming from outside of the cell. Cardiac muscle produces a longer contraction, over 200 milliseconds, compared to the 20 or 40 to 100 milliseconds of our skeletal muscle cell. And so that we cannot tetanize that cardiac muscle, it has a much, much longer refractory period. Lastly, obviously, the muscle action potentials are what determine the contraction of the muscle cells. And remember, when I want muscle cells to work together in my arm, I need a motor unit, one neuron that talks to hundreds of muscle cells so that they all work together. Do I need motor units to get my heart cells to work together?
No.
Why not? How do I get my heart cells to work together then?
They just do.
They use a gap junction?
Yeah, they have the gap junctions that connect them together. So notice our cardiac muscle does not need motor units because as you mentioned, they're interconnected by gap junctions. So those gap junctions allow them to work together. Sending that electrical signal through our heart in just the right way to cause the heart to contract properly to move blood through our body. If you take 431 from me, in fact, the very first organ system we will work on is the cardiovascular system. And we'll talk even more about these cardiac muscle cells and how they work and how they function. And you'll learn everything you ever wanted to know. You're going to learn about it in anybody's 431 class, but in my 431 class, that's what we do first. Cardiovascular.
Is 431 more difficult than 430?
No, it's again, the same way that I teach 430, I continue to teach 431. So the format is the same. I think the only thing that is slightly different is there's a little more physiology in 431, right? Because digestive system, cardiovascular system, respiratory system, there are all these physiological processes that go along with that. And a little less, well, obviously you're gonna have to learn many blood vessels. There's not gonna be quite as much anatomy as like the bones and bone features or the muscles and organs and insertions and actions. So there's a little more physiology, a little bit less anatomy. And so that's really the only difference. But otherwise, it's pretty much the same. All right, questions on this? All right, excellent. And obviously, as we talked about, some of the cardiac muscle cells, well, many of them are involved in contraction. Some of them actually are those ones that are autorhythmic, generate their own action potentials, and allow the heart to beat by itself. All righty. Your book's got some nice tables that review this information and identify the different types and talk about that. So make sure you know all of that. Like I said, clearly we did not go into as much detail on our cardiac and our smooth muscle as we did on our skeletal muscle. But like I said, we'll get a lot more about these in 431. And in fact, the heart, you'll learn everything you ever wanted to know and more about the heart in 431. But that's enough for now. All right, questions on that? All right. Good news is that is everything we need to know from a lecture standpoint for our muscular system. However, we do still have some work to do in lab. We have to finish off our legs as well. So we left off last time with our quadricep femoris, identifying those. And again, remember, this one had an insertion pathway. The only muscle system, muscle group, but the only muscles period that will have an insertion and an insertion pathway. So we left off there. And so that's where we will pick up after the break. But let's go ahead and take our break first. So this is a good transition period. Grab your papers, grab your red pens, grab your blue pens, and we will come back and we will pick up, let's see, it's 9:50 now. So at 10:05, we will restart. All right. Questions on any of that? All right, this might also be a good time to start thinking of and writing down questions for the review, because when we're done with the muscles, then we will go into review mode. So this would be a good time to think about that as well. All right, I'll see you guys in 15 minutes. All righty. Any questions before we dive back in? All right. We finished off last time going through our second muscle group of the thigh region. We did the hamstring and we did the quadriceps, so we got both the big ones. But there are additional muscles we need to talk about. And the next two muscles I want to talk about are the Gracilis and the Sartorius. These muscles are very different, but also very similar to each other. And I think a good way to see this is, again, to start with drawing the origins and the insertions of these. So let me, oops, hold on, insertions, this there, perfect. And all right. Our gracilis is the most medial muscle of the leg. Not surprisingly, as such, it has an incredibly medial origin, as medial of an origin as it can have. So what would be the medial origin of the gracilis?
The linea aspera.
For the gracilis?
Oh, sorry, that was the one above it. The ramus of pubis.
There you go. Right, basically that inferior ramus of the pubis. It's right next to the pubic symphysis, which is right on the midline of the body. So this is indeed about as medial as you can get on the body. We need a nice medial insertion as well. And what's the medial insertion of the gracilis?
The medial condyle of the tibia.
Yeah, notice as we mentioned in the last class, there were not one, not two, not three, but 4 muscles that insert down here into the medial condyle of the tibia. What were the 2 we learned in the last class? Or the 2 muscles from the last class that inserted, there we go, semimembranosus. Membranosus is medial. And if it's a semimembranosus, then what must the other one be? How about the one that's on top of it, the semitendinosus? There you go. Excellent. So those are the first 2. This is the third. And so notice we have this nice medial muscle that starts medial and stays medial and inserts into that medial condyle. All right, excellent. So that is the origin and that is the insertion of this nice medial muscle. Starts medial, stays medial. How many joints does it cross?
2.
2, both the hip and the knee. And how many functions are there for the gracilis?
4.
4. What are they?
Adduct.
Excellent. And I think, hold on, let's stop there. I think that is definitely the most obvious, right? Being a medial muscle, it is going to bring the leg medially. So adducted. What else?
Flex.
All right, excellent. It's going to flex the hip. What else?
Medially rotate the thigh for the hip.
Yeah, let's actually do all of these things to keep them consistent. So it adducts the leg, flexes the leg, or let's actually, here, let's do it with the joint. Leg would be fine, but let's focus on the joints. So it adducts at the hip, it flexes at the hip, it immediately rotates at the hip. Excellent. But remember, it also crosses the knee. So what effect does it have on the knee?
Flexes the knee.
It also flexes the knee. So basically, this is a strap-like muscle on the inside of your leg that basically collapses the leg. It pulls the knee up, it pulls the knee in, and it immediately rotates. So it brings this muscle up and in, flexing both the hip and the knee, bringing the knee up towards your chest, bringing it in towards the midline, and then also twisting it towards the midline as well. All right, and you see this a lot in soccer. Soccer is a big thing here in Sacramento, continues to get bigger and bigger. Everybody's watching Ted Lasso right now. When someone's taking that penalty kick or a corner kick, not a penalty kick, but a corner kick or a free kick, you got all the guys lined up in front. who can't catch the ball with their hands, but also don't want to get hit in the junk. So they're all kind of bent over and twisted in a little bit to try to protect their manliness. And so this gracilis helps in that function. Now, notice I said this gracilis is similar, but also different from the Sartorius. Where does the Sartorius begin? What is the origin of the Sartorius?
The anterior superior iliac spine.
Our Sartorius is actually the longest muscle in our body. Now, does longest necessarily mean strongest?
No.
No, we know diameter means strength, right? So just because it's long doesn't necessarily mean it's strong, but it is indeed the longest muscle of the body. And it has this big lateral starting point up here on the superior anterior iliac spine. Remember, that's right at the top of the front of the iliac crest. So way up there at our hip. But where does it insert?
In the medial condyle of the tibia.
It is the fourth muscle that inserts into this medial, oops, let's make it a little bigger. Here into the medial condyle of the tibia. So this is a muscle that actually starts lateral and then comes across and inserts. And obviously I'm exaggerating this a little bit here, but not so much as you would think. Oh, that's really horrible. So it starts lateral, but it ends medial. Notice this is significant because this strap-like muscle like the gracilis crosses two joints, the knee and the hip. Like the gracilis, it is going to flex the hip and it is going to flex the knee, collapsing the leg down. However, remember, we bring origins towards the insertion. The insertion towards the origin. So since this is a lateral muscle, instead of bringing the leg inward, it is going to bring the leg outward. So instead of adducting, what is this muscle going to do at the hip?
Abduct.
Abduct. And instead of medially rotating the hip, how is it going to rotate the hip?
Laterally.
Excellent. So notice, as I mentioned, these are similar and also different. They have a shared insertion, so they're going to have common effects, but they're also, because they have different origins, very different origins and very different locations, they're going to have very different effects as well. If you remember, this was the example I used at the very beginning of class of crossing your legs, right? If you flex your knee, flex your hip, abduct your hip, and laterally rotate your hip, you're going to put your right foot, if you do this on your right leg, you're going to do this right foot onto your left knee, right? And as long as you're not wearing a skirt, I am, so I can't show you, but as long as you're not wearing a skirt, you can cross your legs appropriately out in public. All right. Notice we've seen the drawings of them here. Let's actually look at the pretty pictures of them. Again, notice here we see that Sartorius. Again, notice it starts lateral, as lateral practically as you can get, right here at the end of the crest of the iliac, the superior anterior iliac spine, and then it comes across medially. In fact, notice when it comes across medially, it actually wraps around the head of the vastus medialis, helping to make that head, which we saw on the bodybuilder right next to the kneecap, prominent. Conversely, our gracilis starts medial and stays medial, coming into that same place. And it is the medialmost muscle of the leg. Got a couple other ways we can see this. We'll get to the rest of these things in a second. Come back to that. Notice here with our bodybuilder. With our bodybuilder, if you look closely, you can actually see this strap-like muscle that is starting up here laterally. If these pesky pants weren't in the way, we could see it going all the way up. But we can see this strap-like muscle that is coming around and wrapping around the head of the vastus medialis. What would that strap-like muscle be?
Sartorius.
Sartorius, absolutely. Where's the gracilis? Do we see the gracilis in here?
Is it that right there? Yeah.
Well, maybe, although really, let me erase that. Do you really see any definition there?
No.
Think about this. This muscle is a muscle. It's a small, thin muscle, obviously, but it's also a muscle. Being a medial muscle, its primary job is to adduct the leg. Now, if I'm standing up and I abduct my leg, do I really need to contract the medial muscles to bring my legs back together?
Not really.
No. What brings my legs back together?
Gravity.
Gravity. I mean, technically, if I was doing a handstand, I'd have to use those medial muscles to bring my legs back together. But do I do a lot of time standing on my head?
No.
No, probably not, right? And even in the gym, there is a machine that will work those adductors of your legs, right? It's that machine that looks like a torture contraption you get in, that machine that you never make eye contact with anybody while you're using it, right? But do you see a lot of people spending a lot of time on that machine?
No.
No.
No. So notice even in our big, huge, massive bodybuilder here, do you see a lot of definition in his adductor muscles?
No.
No. So you're really not going to see it on a bodybuilder. You're not going to be able to see that definition because even our bodybuilders don't work out that muscle. But here, notice with some of the other muscles cut away, we very clearly, very easily, very nicely see that medial most of muscles, that gracilis coming in, starting medially, basically right at the pubic symphysis from that inferior pubic ramus into that medial condyle of the tibia. All right. Questions on the origins, insertions, or actions of either of those two muscles? All right. Notice I said the sartorius is our longest muscle, but it's not our strongest. Remember, we talked about the rectus femoris because of its bipennate shape is one of the strongest muscles in the body, and it needs to be really strong because it needs to move the leg. There is another really strong muscle as well that moves the leg, and it is a muscle group. It is a muscle group made up of 2.5 muscles. The 2.5 muscles that make up this muscle group are the psoas major and the iliacus. Notice the iliacus is located on the inner surface of the ilium, on that iliac fossa. The psoas major is anchored here, attaching to the lumbar vertebrae. Now notice 2 things. Do you need to know the names of these individual muscles?
No.
No. Do you need to know the origins of these individual muscles?
No.
No. But knowing where to find them is vitally important. Now notice I said 2.5 muscles because some people have a third muscle, the psoas minor. Some people, that psoas minor just fuses with the psoas major and you can't really distinguish it. But like I said, whether it's 2 or 3, it doesn't matter because you don't need to know the individual muscles. What you do need to do is be able to find these muscles and think about where they're found. Notice these are muscles that are anchored to some of the most stable structures in our body. We have this iliacus inside the wall of the pelvis. We have these psoas muscles attached to the lumbar vertebrae. Notice these are strong, stable origin points, and they're actually inside the abdominal pelvic cavity. This picture does a great job of showing this, but notice I also had this picture here. Oh, I'll show that in a second. Oh, I thought, where did I have that picture? Oh, I know where it is. It's in the leg pictures on your. All right, so let's come back here for a second. And then we were back here. So. There we go. In your modules, there are the leg model pictures. And notice this is this great picture of the torso. Again, notice we can see where the cut wall of the torso is. And notice if you look really closely at this picture, you can actually see the 1, 2, 3 layers of the abdominal girdle. The outer one, of course, is the external oblique, then the internal oblique, then the transversus abdominis. You can see those 3 regions in the cut side. But here we see that inguinal ligament, which we know goes from the superior anterior iliac spine to the pubic tubercle. And then here inside, we can see in the abdominal pelvic cavity is the iliacus. Here is the psoas major and the psoas minor. And notice all of these muscles come together. They come out the inguinal ligament, and this little bit of it that comes out the inguinal ligament right here, all of the fibers have fused together into a single muscle. And that inserts on the front of the leg. So again, its job being this big, huge, strong, and stable muscle is to lift one of the largest portions of your body. Notice it just crosses one joint, the hip. And so what is the one action of this big, huge, strong, stable muscle?
Flex thigh.
Flex the thigh. Bring the leg up.
So where
Right.
would you point to when, like, if you were asking us, what is this muscle?
So I'd probably
just
have
point?
an arrow like this, probably
Oh, okay.
pointing at both of it on something like the torso like this. But notice also if we go back on our pictures, notice here the leg models in the class. There's the psoas, there's the iliacus. Notice here's the inguinal ligament, and we can even see that little bit of it poking out here as well. So all of those are places that clearly show this muscle really nicely. And notice here's the sartorius. Here's our rectus femoris. So notice this femoral triangle is where the psoas, the iliopsoas, the muscle group that you need to know, comes out. And notice this is also where the femoral artery, the femoral nerve, and the femoral vein are located. For those of you who work in the emergency room or maybe on an ambulance as an EMT or something like that, if someone has a major leg injury that you can't get a tourniquet around to stop the blood loss, what you're supposed to do is palpate that femoral triangle, take the, your palm and basically put the pressure on that to stop the blood flow from coming to the leg to try to save them. Of course, that's all great. You can stop the blood flow, but notice if you push too hard, you can crush that femoral nerve. So now you've saved that person's life, but now they have a limp. And because of the litigious society we live in, even though you saved that person's life, they're still going to sue you. So remember to make sure they sign the waiver before you save anybody's life.
Nice.
All right. Questions on that? All right, excellent. So notice the illustration shows this nicely. Oh, and it even shows the femoral triangle of that iliopsoas, the muscle group made up of the iliacus and the psoas major. Now, notice deep here to the gracilis, and actually I think that other picture shows it really nicely. Notice here, oops, okay. We were talking about the gracilis, and we were talking about the the soccer players. I had a student a few years ago who thought her idea of what you use the gracilis for was her fangirl pose. So notice she took a picture and to share with the class. Notice she is deducting her legs. She is immediately rotating her legs so that the knees come in and the feet come out while she's flexing the hip and she's flexing the knees. So there she is at her Justin Bieber concert, fangirling out using her gracilis. All right. But this was the picture I wanted to show you. Notice there is, in this case, kind of the opposite of what you need to know. There is a muscle group in here, and this muscle group in here is made up of three muscles: the Adductor Brevis, the Adductor Longus, and the Adductor Magnus. Do you need to know this muscle group?
One, the Adductor Magnus.
Yeah, you don't need to know the muscle group. You don't even need to know all three muscles of the muscle group. The only one you are responsible for is the largest of these muscles, and this is the Adductor Magnus. It's the widest, it's the biggest, it's the Adductor Magnus. It is easy to find because notice it is just deep to the gracilis. Notice if we go back to that leg model picture, let's see if we've got a good picture of it. Perfect. Such a great picture. Excellent. All right, let's see here. What muscle is this?
Is it Sartorius?
Sartorius, excellent. What muscle is this?
The Gracilis.
Gracilis, excellent. Notice also back here, also inserting to the same part, is this deeper, wider muscle and this narrower top muscle. What are those two muscles? What's the deep muscle?
The semimembranosus.
Yep, semimembranosus. And what's the superficial one on top?
Semitendinosus.
Semitendinosus. Excellent. Perfect. All right. Notice we can even see where the iliopsoas comes in a little bit right here. All right. But the reason we're looking at this is notice here's our gracilis, that superficial muscle. And remember, as we talked about, just deep to it is our Adductor Magnus. So we have that Adductor Magnus, which is going to be that deep muscle. Notice if we go here, right here, if we go to that superficial picture. So again, notice right there is our Adductor Magnus right beneath the gracilis, and it's wide, so you see it on the front and on the back. So on the lateral aspect of the leg, you'll see the front and the back of that Adductor Magnus. Now, the Adductor Magnus, you don't need to know the origins and the insertions, but how many actions does it have?
3.
All right.
Okay.
Being an adductor, what do you think its most obvious action is?
Adduct.
Adducting the leg. Excellent. What are the other two actions?
Immediately
Immediately
rotate.
rotate.
Flex thigh.
Yep, there you go. So it flexes the thigh, immediately rotates the thigh, and adducts the thigh. Brings it all in. It's that nice roundhouse kick that this Adductor Magnus does for us. Now, being primarily an adductor, as we talked about, there aren't a lot of exercises because gravity does most of its work. So it's a muscle that doesn't quite have the muscle tone that a lot of other muscles do. However, if you happen to be a soccer player or a running back in football or something along those lines, you often hear people who have to make a lot of twisting and turning types of motions that someone is out for 2 weeks because of a groin injury. When they say groin injury, is that really having anything to do with the male reproductive organs?
No.
No. 80% of groin injuries, when you have an athlete who strains a muscle that they collectively refer to as the groin injury, 80% of those involve straining this Adductor Magnus muscle. Because this is just a muscle, like I said, because of its location, because of its action, doesn't get a lot of work. And so it doesn't have the tone of many of the other leg muscles. So when you hear about an athlete getting a groin injury, it's 80% of the time this Adductor Magnus muscle. All right, questions on that? I believe we just have one more muscle then of the thigh, and it is the smallest. And like a lot of these, they have to compensate, so it has the biggest name. Tensor fascia latium. Notice again, you don't need to know the origin and the insertion of it, but notice it does connect up here to the superior anterior iliac spine next to the sartorius. They form the V at the top of our rectus femoris. And not surprising, with a name like the fascia latium, it inserts into that iliotibial band. This is the muscle that, as we mentioned, its primary function is to keep tension on the IT band, which keeps tension on that connective tissue. Notice your illustrator for your book has put a small strip of that fascia lata around the leg. We looked at the cadaver and saw it. And so it acts as that pressure cuff to help to stabilize the muscles and to bring the blood back up to the heart. However, it is an accessory muscle, but the good news is while its name is challenging, its function isn't. This is an anterior muscle. So as an anterior muscle, what does it do to the hip?
It flexes.
And it is a lateral muscle. So as a lateral muscle, what does it do to the hip?
It laterally rotates.
Well, not laterally rotates, but it laterally brings it out to the side, which
Adduct.
is
Oh, it abduct.
abduct.
Yeah.
There you go. So it flexes and abducts the leg. All right. Questions on that? Again, there's that cut section of the fascia lata. We did that, we did that, we did that, we did that. Excellent. With that, we are now done with the thigh, and we can come down here to the lower leg. There are a couple muscles of the lower leg we need to know the origins and insertions of. Remember, we were a little tricked with the bicep femoris. Its name is very similar to the bicep brachia, but it didn't have the same types of origins and insertions. The two muscle bellies laid on top of each other. One was superficial and one was deep. Whereas here is what we commonly call the calf muscle, is a muscle that is the gastrocnemius, that is almost identical in shape and function to the bicep brachia. Remember, the bicep brachia had two bellies side by side. They crossed two joints, both the shoulder and the elbow. And even though the bicep brachia sits on top of the humerus, did it actually attach to the humerus?
No.
No. Remember, both of its origins were up on the scapula, and its insertion was down on the radius. So even though it sat on top of the humerus, it didn't actually attach to the humerus. Oh, and of course, it's superficial. And it's the one that gets all the press. We talked about doing the curls for the girls, right? To get that nice big prominent bicep muscle. Well, it's the same thing with the calves, right? If you don't like your calves, you can actually get calf implants. In ancient times, and by ancient times, I mean the 90s, there was a football player, a wide receiver for the Cincinnati Bengals by the name of Chad Johnson. Chad Johnson at the time was probably the fastest wide receiver in the game. He once challenged a racehorse to a 100-yard dash, and he beat the racehorse.
What?
He was known for his massive calf muscles. And in fact, an implant company gave him at the time $500,000 to take a mold of his calves so they could make implants so that if you didn't have calves like Chad Johnson, you could get these implants so that your calves would look like his. Of course, a lot of this fame and popularity went to his mind. You may not know him as Chad Johnson, but maybe you may know him by what he changed his name to. His number with the Cincinnati Bengals was 85, and he changed his name to Ocho Cinco and had that on the back of his jersey for a while. But again, very, very famous player with massive superficial gastrocnemius muscles. So much like the bicep brachia gets a lot of attention, so does the gastrocnemius. Now, let's see how this relates. We have, we'll just do it over here. Excellent. How many bellies do we have for the gastrocnemius?
2.
All right, excellent. Side by side bellies. And how do we distinguish them?
Medial and lateral.
Excellent. How do we know which is which?
Medial side and lateral side.
How do we know which is medial and which is lateral?
Posterior.
Medial is
Tibia and
acquired tibia.
There you go. The tibia is medial. So on the tibial side, we would have the medial head. And on the lateral side, on the fibula side, we would have the lateral head. What is the origin of the medial head?
Medial condyle of the femur.
Excellent. Now, do you think it's going to be on the round knuckle part that forms the joint with the tibia and then rotates and gets pinched in there?
No.
No. So where it's actually located is up here, right above the knuckle. If you look on the femur, right above the knuckle, there's a little indentation there. So notice we're still on the medial condyle, we're just above the knuckle. And so that is the origin for the medial head. And of course, what is the origin for the lateral head?
The lateral
Sarah's
condyle.
got it. Exactly. It's the exact same thing. That same notch above the knuckle on the lateral side. So notice, even though it sits on top of the tibia and fibula, it does not originate from the tibia or the fibula. And what is their shared insertion?
The calcaneus.
Yeah, that heel bone. So notice their insertion is not on the tibia or the fibula either. Instead, they have a shared tendon that comes in and inserts. And in fact, even though I've drawn this really wide, this tendon is the biggest and strongest tendon in the body with the largest amount of tension in it. Hopefully nobody here has torn this tendon. What is this tendon called?
Achilles tendon.
Yeah, it's the calcaneal tendon because it inserts into the calcaneus, that heel bone, but also it is such a ubiquitous term, I will accept Achilles tendon for the name of this. Of course, why is it called the Achilles tendon?
You got me there. I don't know.
None of you know?
No.
Is it the Achilles heel? Is that why?
Yeah, but where does the term Achilles heel come from?
Oh.
A Greek thing or something?
It is indeed a Greek thing. Depending on which version of the Battle of Troy, the Ilium that you read, either his mother, who was a water nymph, so Achilles was a great warrior who was a demigod, half human, half god, because his mom was a water nymph, and to protect him, she either, depending on which version, dipped him into the river Styx or dipped him into fire to basically make his skin impenetrable to mortal weapons. However, to hold him, to do that, to immerse him, she had to hold him by his heel, and so his heel was his one weak point. So during the Battle of Troy, he got shot with an arrow in the heel and died as a result of it. So that is where the term Achilles tendon comes from. Hopefully none of you have torn your Achilles tendon, but has anyone ever been around someone when they tore their Achilles tendon?
Yeah, my sister did.
Were you nearby when it happened?
Yeah.
Did you hear it?
Yeah, it was loud.
Yeah, when you talk to people who tear their Achilles tendon,
like really loud.
You ask them, and often they feel or they think they've been shot because there is this loud popping noise, and then you're collapsed to the ground. It can be heard up to a half a mile away. There's so much tension if it tears completely. There's so much tension that when it rips, it is incredibly loud. It rolls up and balls up the muscle into the back of the leg. It can be an incredibly brutal injury when it occurs. And as Ava, as I'm sure you know, it can take years to recover from. And some people never fully 100% recover from it. Because it is such a challenging. Again, remember, this is a big, huge tendon. And tendons, as we know, have parallel collagen fibers. So when you damage it by tearing it longitudinally across that, scar tissue forms. It can't hold as much tension. It can be very, very challenging to recover from. Excellent. Did your sister recover from it?
I mean, kind of. I think it still hurts her. She went to physical therapy for a really long time.
Yeah.
So.
Well, I'm glad it is better. So yeah, absolutely. Hopefully, eventually it will get back to the way it was before. All right. So there, as you can see, is something very, very similar to our bicep brachia.
I have a question.
Yeah, go ahead.
Quick. When it comes to the insertion, on our paper, it says the calcaneus via the calcaneal tendon. Would it be acceptable just to say the calcaneal tendon?
No, the insertion would be the calcaneus.
The calcaneus. Okay.
Yeah, the structure would be the calcaneal tendon. If you said calcaneus via the calcaneal tendon, I would accept that. But remember, an insertion is a attachment point. What you kind of described was an insertion pathway, but remember, I'm not holding you responsible for a second one of those. So make sure insertion is where it attaches. So make sure you give me that calcaneus. You can say or not say calcaneal tendon. I don't care either way for that part, but make sure you say the calcaneus.
Okay.
Now, conversely, on the exam, if I have an arrow pointing here, an arrow pointing here, and I ask you to identify that structure, then that's something different. Then I specifically want calcaneal tendon or Achilles tendon. Like I said, I would accept both of those. But if I have it here and I ask for the insertion, or if I have it here and ask for the insertion, make sure you see the calcaneus.
Okay.
Okay. And of course, I would also point it here and say, identify the muscle B specific. And what would your answer to that be?
The medial head of the gastrocnemius.
Perfect. Absolutely. Now, how many joints does the gastrocnemius cross?
Two.
Yeah, like the bicep brachia, it crosses two. Being a posterior muscle, what effect does it have on the knee?
Flexes.
Flexes. Now, when we haven't talked about the ankle yet, but or at least none of the muscles that affect the ankle, but let's see, do I have a side view? Let's take a look here. Perfect. As we learned in the skeletal system, the ankle joint is really formed by the C-clamp that is formed by the medial malleolus and lateral malleolus of the ankle, of the lower leg. So when muscles come in and insert behind the malleoluses, then when they contract, they're going to pull the back of the foot up, which would point the bottom, the top front of the foot down. And what type of action would that be?
Plantar flexion.
Yeah, we're flexing the plantar surface of the foot. Conversely, if a tendon comes in and inserts in front of the malleoli, then when it contracts, it's going to bring the front of the foot up. And what would we call that action?
Dorsiflexion.
Dorsiflexion, flexing the dorsal surface of the foot. So our gastrocnemius, which we see a little bit of here, here's the lateral belly of the gastrocnemius. And since it's coming in and inserts into the calcaneus behind the medial and lateral malleolus, what is the second action of our gastrocnemius?
Plantar flexion of foot.
Yeah, so it flexes the knee and plantar flexes the foot.
When
What would you, go ahead.
When we answer that, can we just put plantar flexion? Because the foot, I mean, really.
Great question. Okay, here's my answer to that. You are correct. Plantar flexion and dorsiflexion are actions that solely occur in the foot. Kind of like supination and pronation is just in the forearm.
Yes.
So if you just put plantar flexion, it would be correct and I would give you full credit for it. However, it is probably a good practice to get used to not just saying the action, but saying either the joint or the part of the body that moves. So while it is completely accurate to say the gastrocnemius flexes the knee and plantar flexes, you would get full credit for saying that. If you said flex and plantar flex, you wouldn't get full credit for that because you haven't told me what you flexed. So it's probably a good habit to always give me the action in the joint or the action in the part of the body. So as good practice, it's probably best to say plantar flexion of the foot. But to answer your question, no, I would not mark it wrong if you just put plantar flexion.
Okay.
Okay.
When you ask us for the actions, let's say we get, I mean, is it graded just by if you get some of them right? It's half credit, and if you don't get all of them,
yeah.
Like,
Again,
how is that graded?
I'm not slicing this any thinner than half points. So
Right.
even if something has 4 actions, if you get one of those actions correct, then you'll get half credit. So if 3 of your actions are wrong and one of them correct, you'll get half credit, right? But it works the other way as well. If something has 4 actions and you get 3 of them right and only one of them wrong, you're still getting half credit. So yeah, I'm not gonna be slicing this
Right.
quarter points or 3/4 points or 2/5 of a point. That's ridiculous. So I don't cut it any slice. So yes, as long as you get, so if you get nothing right, you get 0. If you get all of it right, you get full credit. If you get partial, whether that partial is one thing or all of it except one thing, you get half credit. Okay. So that's how it gets split. What do you use your gastrocnemius for? I mention it because it's that magical time of year. Halloween is now a couple days old. So of course, in our house, You know, Christmas has
Christmas.
exploded. Absolutely.
Is that
True,
when
but
you-
remember, standing on the toes is the plantar flex, but that doesn't flex the knee.
That's just the
As-
true love's kiss thing.
There you
Yeah.
go. Exactly. Absolutely. Right. Again, being Christmas and being in a house filled with women. I have 2 daughters. I have a wife. I have 2 female dogs, and we now have a female cat. So, not surprisingly, this time of year, the Hallmark Channel with all the Hallmark Christmas movies is on 24/7. And how do you know that they found true love at the end of the movie?
They raise their leg.
Mistletoe.
There you go, that true love kiss, right? Where you bend the knee and point the toe. And so every hour and a half, like clockwork on TV, right? The Hallmark Channel, someone's using their gastrocnemius to show that they will live happily ever after. All right. Now, there is one more way that the gastrocnemius is like the bicep brachia. And that is the same way that, even though it doesn't get any credit, right underneath the bicep brachia is the brachialis that helps in the flexing of the flexing of the arm. Well, the same thing is true here. Notice folded up in this space, let's pick a color that'll stand out, is this big flat muscle. Notice it's a long flat muscle, but it's folded up inside of this small compartment, and it is deep to the gastrocnemius. Notice with the gastrocnemius here, we don't really see it, although we do see a little bit of it sneaking out behind, deep to the gastrocnemius. And then it actually fuses and uses the same tendon as the gastrocnemius. So here it's been cut, but there's this broad, flat muscle, which is actually where it gets its name. What muscle is this? Soleus, absolutely.
Yeah.
So solefish is a big flat fish that kind of skims along the bottom of the ocean. And so it's a broad, flat fish. Well, this is a broad, flat muscle, and that's where it gets its name. And it helps the gastrocnemius, but is deep to it and doesn't get any of the credit. Also, like the brachialis, it only crosses one joint. So what is the origin of the soleus?
Head and proximal shaft of the fibula and superior tibia.
Yeah. So again, notice the head and proximal shaft of the tibia, and then the proximal shaft of the tibia. Remember, as we just saw, it's this kind of U-shape origin, which is how we cram that flat muscle into such a narrow space. And where does it insert?
In the calcaneus.
Yeah, it has the exact same insertion as the gastrocnemius. It fuses with its tendon, and that one tendon, that Achilles tendon, does all of that. However, while it's sitting right underneath the gastrocnemius, notice it only crosses one joint. So how many actions does the soleus have?
One.
And what is that?
The plantar flexion of the foot.
Yeah. So this one, Sarah, would be that standing on your toes. This one helps you to stand on your toes because this one does not flex the knee. So this one just makes you look taller in the family photo. All right. Excellent. One more muscle we need to know the origin and insertion of. And let's go here to the pretty picture to see this. I don't remember which picture it is, but I think we can use any of these. Here, this one's a little closer. Excellent. All right. So let's think about this for a second. Here on the tibia, how many muscles insert into the tibial tuberosity? 4. What collectively are those 4 muscles known as?
The quadriceps.
Quadriceps femoris, excellent. How many muscles insert into the medial condyle of the tibia?
4.
4, right? Sartorius, Gracilis, semimembranosus, and semitendinosus. Notice that doesn't leave a lot of room for any other muscles in that region. So while this muscle is called the tibialis anterior, where does its origin have to be?
Lateral.
Yeah, so notice it starts over here lateral, kind of on the lateral condyle of the tibia over here, kind of towards the front, but still a little bit lateral. But where does it insert?
The medial cuneiform and the first metatarsal bone.
Yeah, actually on the underside of the medial cuneiform and the proximal phalanx 1. So this muscle actually comes across the anterior face of the lower leg, and then, I'm exaggerating here, inserts underneath the medial side. And that's what puts it on the anterior surface of the leg. You may not have thought of it in those terms, but I know you're absolutely aware of it, because notice when we look on the anterior compartment of the lower leg, we see a whole lot of the diaphysis of the tibia, which, as you know, hurts like heck when you slam it into the coffee table. And what do we collectively call that?
The shin.
Your shin, absolutely. So we have that very few muscles here on the anterior part, but we do have this tibialis anterior, which starts lateral, but then comes medial and hooks onto the underside of the foot. This is important when we think of its actions. Of course, it comes in front of the medial and lateral malleoluses. So what's the obvious action on the foot going to be? Dorsiflex, absolutely. And Sarah, assume, again, I'm not going to put my foot up to the screen, but assume this was my left foot. Because the tendon comes and inserts on the bottom, not only does it dorsiflex, bringing the toes up, but it also can rotate the sole of the foot so that it points inward. Of course, we don't call that an inward rotation. As Sarah pointed out, we call that the I inversion of the foot. So it I inverts the foot and it dorsiflexes the foot. Excellent. You may not have thought of the tibialis anterior before, but many of you are aware of it because, as we've talked about, there are fascias that wrap around and stabilize our muscles in place. And normally, when those muscles, multiple muscles, are bundled together, they are stabilized, but they also have some flexibility of space to move. However, here on the front of the leg, there's not a lot of muscles. So really, the tibialis anterior kind of has its own fascia that wraps around it. Fascias, as we know, are made of dense, irregular connective tissues. And do dense, irregular connective tissues stretch and expand, and are they elastic?
No.
No. But as we know, muscles are very dynamic. They can get an increase in blood flow. They can swell, they can enlarge from use. And if that muscle tries to expand and it's restricted in a fascia that doesn't allow it to expand, that muscle can become congested. And we have a hard time getting oxygen and nutrients to it, which means it's going to produce more lactic acid. And because it's congested, it can't get that lactic acid out of there. And suddenly, as you're using that muscle becomes incredibly painful, feeling almost like it wants to tear off the bone. And what do we call that? There you go. Mitch has got it. Shin splints. The tibialis anterior, if that fascia is too tight, is the primary cause of shin splints. So what do you do to help to eliminate it? You try to do a lot more stretching, try to loosen that connective tissue first prior to running so that there's more room for the muscle to expand. Right? For some people, no amount of stretching can be enough to eliminate that. And if you're, for instance, a professional soccer player, being in a constant state of having shin splints isn't something that's very, very useful. So in some extreme, they're typically not going to do this for a common person like you or I. But sometimes for professional athletes, they will actually make an incision in that fascia to help to try to loosen it up, to help to eliminate those shin splint issues for some of these professional athletes that have it as a chronic issue. All right. Questions on that? All right. Notice that tibialis anterior is easy to find because it's right here, right on the front, which is convenient because the other two muscles we need to know on the lower leg are right next to it. Notice the next one, we'll use light blue for this, has a small belly right next to the tibialis anterior. But as we've also talked about, what you really need to pay attention to is the tendon. And notice this has a long tendon. And not only does it have a long tendon, but it has tendons that it branches into multiple tendons that go to digits 2, 3, 4, and in this case, even 5. What does that sound an awful lot like? Didn't we have a muscle here whose tendon branch that went to multiple fingers?
The extensor digitorum.
Extensor digitorum, absolutely. This is a very similar muscle in similar function, and so guess what it's called?
Extensor digitorum longus.
Right. It is the extensor digitorum, but because it has to get all the way out to the toes of the foot, it has a longer tendon. So it's the extensor digitorum longus. And the way I remember it is extensor digitorum in the arm, extensor digitorum longest in the leg. So the extensor digitorum longus is in the leg. So don't get those two mixed up. This is not the extensor digitorum longus. This is the extensor digitorum. This one here is not the extensor digitorum. It is the extensor digitorum longus. Longus in the leg. What are its actions?
Dorsiflexion of the foot and extend phalanges.
Yeah, it comes in front of the ankle joint, so it's going to bring the foot up and it's going to extend the toes. So, tibialis anterior, extensor digitoris longus, and then notice there is a third muscle over here that we can see a little bit of, but notice its name, Fibularis longus. It is a lateral muscle, so notice, not surprisingly, we see it best on the lateral side. Notice here we see it on the lateral side. Here we see, again, the extensor digitorum longus. And a little bit of the tibialis anterior here from the front. So we see those three side by side by side. But what we care about, so let's get rid of that and that and that, is this, let's cheat, doop, doop, Fibularis longus. As its name indicates, fibularis, lateral side of the leg, longus, long tendon. And notice this tendon goes behind the lateral malleolus and inserts on the lateral side of the foot. So again, if this is my right foot, this tendon comes down this side in front of the joint and connects to the lateral side of the foot. So what are the two actions on the foot?
Plantar flexion and then the eversion
Yeah,
of the
so
foot.
dorsiflex and eversion. Right? Rotate the sole so that it points outward and bring the toes up. So dorsiflexion and eversion of the foot.
On the list, it says plantar flexion.
Oh, I'm sorry. You're right. It does. It goes behind. I'm sorry. Because it goes
Right.
behind. I'm sorry. Plantar flexion and eversion. Sorry. You are correct. Plantar flexion and eversion because it goes behind. I just finished saying that, but unfortunately, I wasn't listening to what I was saying. But there you go. So behind. So it plantar flexes and everts the foot. And there you go. Just that easily. Those are all the muscles that you need to know the actions, origins, and insertions of. And that is all the information you are going to be responsible for on this next exam. All right, here's the game plan. It's 11:07. Let's take a quick 10-minute break, quick biology break, or get some more caffeine or something along those lines. Gather your questions together. So let us meet back here at 11:17. For our question and answer review for the exam. All right, any questions? All right, quick biology break, quick grabbing some caffeine, and then we will be back here in 10 minutes. All righty. I am done, but it is on you guys now. So I'm happy to answer any questions. That will help you to be successful on your exam on Thursday. Remember, if you don't ask questions, I assume you've all mastered the material and I make the exam harder. Of course, if you don't get asked questions, then I guess we all get to leave early and go to bed. Okay, yes. So the easiest way to demonstrate this is, again, to think in terms of, well, here, my camping flashlight. So if you think of the humerus and you forget what the forearm is doing, but by bending your forearm, you're best able to see this. Notice that when I bring my hand forward, if you think of what's going on in the humerus, is that my humerus is rotating this way. Whereas if I start here and I bring it inward, my humerus is rotating this way. And so both of those rotations, so I am rotating, rotating, right? So this is the rotation. Now, in determining what the rotation is, you always think of what the anterior side is doing. So if I'm doing that, then the anterior side is moving away from the midline, and that is a lateral rotation. If I'm doing this, then the anterior side is going towards the midline, and that is a medial rotation. So remember, rotation is along the longitudinal axis. And when it's a structure that's not on the midline, if it's just on the midline, like the head, it's a rotation. But if it's something that's not on the midline, then if the anterior side is going towards the midline, it's medial. If the anterior side is going away from the midline, it is lateral. And the hip can also be done that way as well. So it works the exact same way with the hip. Yep. So when we make a muscle cell, remember, a muscle cell is made up of hundreds of immature myoblasts that are these stem cells that basically fuse together to form one single cell. As we know, a skeletal muscle cell is surrounded by, let's do it this way, areolar connective tissue, that is that endomysium that surrounds it and insulates it. Well, in those hundreds of myoblasts that fuse together to form that muscle cell, it turns out not all of them fuse to the myoblast. So nestled in here, right next to the muscle cell, inside the endomysium, so right there, right next to it, is the satellite cell. And so what happens is if part of my muscle cell gets damaged or destroyed, then this cell is capable of dividing to produce a whole bunch of new myoblasts, and those new myoblasts will fuse with the old cell, and we're able to repair that damage. So remember, we talked about limited regeneration. This is where that limited regeneration comes in. Notice if a shark comes and takes a big, huge chunk out of my arm, not only are the muscle cells gone, but the myoblasts are gone as well. So I'm not going to be able to repair individual muscle cells if those muscle cells are gone. And because this satellite cell is located within the endomysium, it can help this cell recover, but can it help a cell in the next fascicle, one fascicle over? Is it going to be able to help it to repair? No. So that's why, because it's so closely and intimately related to that muscle cell, it can only help that muscle repair, but it can't help others. And that's why the regeneration is so limited. Yes. The short answer to that is yes, there is creatine available in both of them. Notice it's not going to be it's not going to be as important in smooth muscle because of the smooth muscle's contraction. Cardiac muscle actually has a very special type of creatine, but yeah, cardiac muscle is hyper sensitive to oxygen deprivation. It solely relies on aerobic respiration. And so if deprived of oxygen for even a brief period of time, if it has to produce it anaerobically, make lactic acid, it can be damaged and it can't regenerate. So having extra creatine phosphate or having a special creatine phosphate in the cardiac muscle is absolutely important to make sure that we don't have to make ATP anaerobically. And like I said, we'll talk a lot more about cardiac muscle and even more about smooth muscle when we get to, for instance, like the digestive system in 431 or the cardiovascular system in 431 and stuff like that. So we're introduced to it here. We will definitely get in more depth on those things when we get to 431. All right, what else? I guess the only modification we talked about is, and I'm going to cheat, let's just pretend that this is the myosin now. We know that that power stroke of the contractile cycle has 4 steps, right? It is going to grab onto the actin, perform its power stroke, let go, and reprime. And I think the only real, I'm not even sure I would call it a modification, but the only really thing that we talked about is this situation where what happens if the myosin head can't let go? And what allows the myosin head to let go?
The ATP.
ATP. So the only way it wouldn't be able to let go is if there was no ATP. And what condition is the body in if there's no ATP? Not a trick question. The muscle cell's not making ATP anymore. What's the condition of the body?
Dead.
Yeah, exactly. And so what did we call that condition where the myosin heads grab and pull but can't let go?
Rigor
Mortis. Exactly. I'm not sure that qualifies as a modification, right? I mean, I guess technically death is a modification of the body, but not one you can typically recover from. But I guess that would be the one way we talked about a disruption in the cycle. So I think that's probably the closest to something like that. Yeah. So you need to know what rigor mortis is and what causes it. But I don't think I would, you know, I wouldn't say that that's an essay question of describe a modification of the contractile cycle. I certainly wouldn't ask the question that way. All
Can
right.
you please give examples of the isotonic contractions and the isometric contraction?
Yes. So an isotonic contraction is, again, when the muscle changes length, right? And the key to an isotonic contraction is that the tension remains the same. Notice the weight of what I have to lift stays the same, whether it's here, whether it's here, whether it's here, or whether it's here. So as I am using my bicep brachia, shortening my bicep brachia to lift this weight, that would be an example of an isotonic contraction. All right. Now, if I was holding my arm down, notice I could just relax the muscle and my phone would fall. But I don't want my phone to fall. So I want to lower it slowly. To lower it slowly, again, I have to put a consistent amount of tension in my muscle because, again, the weight isn't changing. But in this case, the tension I'm producing has to be less than the load. To lift it, the tension has to be greater than the load. So to lower it, the tension has to be less than a load. So for that isotonic contractions, there's two types. One where the tension is more than the load. And what do we call that type of isotonic contraction? Concentric. And one where the tension is less than the load and the muscle gets longer. And what do we call that one?
Eccentric.
Eccentric. And notice the key with an isotonic contraction is the load and the force can never be equal. Because what happens when the load and the force are equal?
It's isometric.
Yeah, then at that point, it doesn't change length. At this point, the amount of force that I'm putting into my hand, my elbow, is the exact same force of the weight of the load. Now notice I could add more things to my hand, and does my hand suddenly fall down because of that?
No.
No. If I stayed at the same tension, it would, but instead I have the ability to add more tension. And so as long as I add more things and add more tension, and as long as the two are always equal, then I will get no change in length. All right.
Thank you.
Yep, no problem. And I saw that there was another question in here as well. Great question. So you don't have to worry about so much the binding of it so much, but the key is to think of it this way. We want our sarcoplasmic reticulum to hold as much calcium as possible. Remember, as we said, outside of the matrix of our bones, the sarcoplasmic reticulum of our skeletal muscle has the highest concentration of calcium, second only to the bones. So, right, you've got 2 whole days before the exam, perfect opportunity to take a 2-day vacation, right? Go to Vegas for 2 days because, of course, you'll get a lot of studying done in Vegas. And when you grab your carry-on bag to go onto the plane to go to Vegas, do you just randomly throw as much crap as you can in there?
No.
Your clothes and your all those things, do you just randomly throw them in there? Can you fit as much stuff in there if you just randomly throw them in? No, you want to tightly organize them and pack them. And if you can tightly organize and pack them, you can get more stuff in your bag. And that's basically all salquestrin does. Calsequestrin is a protein that basically helps to organize the calcium inside here so we can get more in there. So it's just a special protein that helps to organize the calcium to give us, because if it's more organized, we can get more calcium in there. So basically, it's just an organizing protein that helps us to get more in there. Yeah, I could. So not on the lab exam. So just for clarification, on the lab exam, I will specifically ask, right, what are the 1 action, 2 action, 3 action, 4 actions of this muscle? So I'll point at a muscle and ask that. However, in the lecture exam, like in a multiple choice question, could I ask a multiple choice question that would say, for instance, which of these muscles would allow you to do a jumping jack? And if I gave you the choice of the deltoid, the masseter, the adductor magnus, and the rectus abdominis, then which one is going to allow us to do a jumping jack? Right. Clearly, it's the deltoid. Absolutely. Right. Or could I say paralysis of which muscle would not allow you to flex the hip? Right. And then I had some listed that way or something along those lines. So sure, those kind of questions could be the kind of things that could show up in a multiple choice question, or maybe one of the fill in the blank types of questions or something along those lines. I don't think it's quite essay question worthy. But and like I said, and it won't be on the lab exam. The lab exam is all about origins, insertions, actions, and well, names for that matter. And then of course structures. But yeah, thinking of what a common activity might be and what muscle would do it, that's the kind of thing that I think would be a reasonable question on the lecture exam.
So for the lab, it's going to be, you said about 30% of bones, and then
I think 30 is a little high. No, I would say
okay.
I would say probably somewhere between 10 and 20% bones. Again, I haven't finalized it, but so let's do some math. This question on average usually has somewhere around, let's say, 85 questions. Although the math on that's going to be hard. Let's say 80 questions. So maybe, I don't know, 10 to 15, something like that, would be bone questions. Maybe, well, somewhere around 2 to 5 probably would be bodybuilders.
And like histology wise.
Yeah, I was trying to think histology wise, because histology, right? We have obviously the microscope stuff, but we also have the sarcomere model. We have those 2 models of the muscle cells, skeletal muscle cell, I should say. For the microscope, remember, we also have the 3 muscle type models. The smooth muscle, the cardiac muscle, and the skeletal muscle. And remember, for all of those, I could, just like we did today in class, I could show you the smooth muscle model. And could I ask you characteristics?
Yes.
Of that particular muscle tissue type? Sure, absolutely. Sarcomere model, skeletal muscle model, models, plural, there's 2 of those. I guess, I'm not sure whether you'd put it here with the skeletal muscles or you'd put it up here with the bones. There's that great rotator cuff model that is the scapula with the muscles on it. So you could either count that as a bone picture or maybe as a skeletal muscle model picture, because obviously there's going to be, and then obviously the majority of it is going to be models of, all right, the arms, legs, trunk, head. And again, along those lines, there'll also be, you know, the charts or illustrations. And again, for those, basically, they're the 4 main things you need to know. And those are going to be the origins, the insertions, the actions, and the names. And then, of course, also remember anything that has multiple heads, you need to be specific, which will be the fancy term you'll say. And that will be for origins and names of the two-headed muscles.
So how many questions for like the histology stuff?
I don't know. It's hard to break it down. There's a lot of that. So, I mean, there's definitely going to be, it's not going to be 50-50. So there's
Right.
definitely going to be, hold on, more of this than of that, but I don't know exactly how much. I don't know. Like I said, that math's getting a little too fine. So,
Well, my more specific question was like, is the O's, I's, and A's like evenly split up? Like, are we going to get a bunch of picture? Like, I know it's in a
Well, okay.
database, but
I have no way of knowing.
oh, okay.
Because if I was writing the exam, then I might be able to more evenly distribute between origins, insertions, and actions. However, these things are all in test banks, and every single one of you is getting a completely different exam. So is every single one of you going to get the same number of origins and the same number of insertions and the same number of actions?
No.
No, by random choice, you may get 3 times the number of origins that you do insertions, because it's just going to be random, right? Or you may get more names and less actions. That's the problem with this format, is that everybody's getting a different test, so everybody's is going to be different. So the distribution of the origins, insertions, and actions and names, for that matter, are completely random. So
Okay.
you may have, you know, so how those 4 break down. And again, and that's, I'm fine with that, because all 4 of these I think of as even types of information. You need to know all 4 of these things for all of them. Now, the one thing I will say is, obviously you don't need to know the origins and insertions of all muscles. So are there going to be more names and be more actions? Yes, just simply because you don't need to know all the origins and all the insertions of all the muscles, but you do need to know the names and actions of all of them. So yeah, there'll probably be more names and actions, but how it's going to break out, it's impossible for me to know.
Right. Okay. So then in person, is it just a little bit easier just because you're able to, I guess, specify more even for like essay question wise, what's going to be on the exam, since everyone has the same one?
Yes and no. There are definitely questions I like more than others. So I purposely, when I write my tests, I do randomly assign the essay questions, but I do get a chance to double check. So if I feel it's too loaded one way or another, or, you know, emphasizing one concept or more than another than it should, then I can make those adjustments, but that's nothing I can do in this. But that's been the truth for all of our tests so far.
Right.
So again, that's nothing new. That's the problem with this online format. Yeah. Yeah, absolutely. So the key with lactic acid is we get lactic acid from the breakdown of our glucose, gets broken down into pyruvic acid, so all the time in every muscle cell, glucose is broken down into pyruvic acid. This process is called glycolysis. And again, as we talked about, this produces 2 ATP. When oxygen is present, this pyruvic acid enters the mitochondria. And when it gets into the mitochondria, it's broken down completely. And from this, we get lots of ATP, 32 to 36 net per glucose. However, as we also mentioned, if this muscle is working really, really hard without oxygen, that pyruvic acid is converted into lactic acid. And so lactic acid, as we talked about, is toxic, is damaging, changes the pH, and it is one of the primary factors that leads to fatigue. So during recovery, we need to be able to get rid of this lactic acid. Obviously, one of the quickest and easiest ways to do that is to release it into the blood and send it to the kidney. When we send it to the kidney, then obviously we pee it out. And that is a useful way of getting rid of it very quickly. The problem is lactic acid still is a potentially valuable resource. And that's because with oxygen, we can actually convert it back to pyruvic acid. Oops. So in the presence of oxygen, if we can get more oxygen into the cell, we can take this lactic acid and we can convert it back into pyruvic acid. And then, again, as we just talked about, if we have enough oxygen to convert it back into pyruvic acid, then we probably have enough oxygen to then have it enter the mitochondria and be broken down to produce ATP. And so we can use the ATP from the lactic acid to do the work of recovery. Right? Repairing the proteins, pumping ions where they need to go, right? Doing all the things, all the work that needs to be done, making the new proteins that are going to be necessary to repair or add myosin, realign the proteins in the sarcomeres, all the things that go along with that. Now, as we mentioned, since it is, so again, we can break it down ourselves or recycle in the muscle cell. But does that mean that any of that lactic acid that was released into the blood is just automatically lost and we're never going to be able to use the resources of it? No. So while some of it is in the blood, will be released in the kidneys, the other advantage of getting it into the blood is we can get it to the liver. And so the other place where we can recycle is in the liver cells. So just because it leaves the cell into the blood doesn't mean it's lost. Yes, some of it will leave via the kidney, but the same way that the muscle cell is converting it back into pyruvic acid and using it to make ATP, the liver can do that as well. And as we know, the liver stores glucose, so there's even a limited ability of once we convert it to pyruvic acid, we may actually be able to get it to go back together into glucose and store it in the liver as well.
Can we compare the skeletal muscle action potentials and twitches to the cardiac in like a chart?
Sure. All right. Let's start with ways that they are similar. What are the ways that skeletal muscle cells are similar? As skeletal and cardiac muscles are similar in their muscle action potentials. Actually, here, let's do this way. Excellent. Oops. So they have, both have thresholds. Negative 60 millivolts. Excellent. What else? Okay, so we want to be careful. We're talking about the muscle action potential here, but if we want to throw the twitches in here, we can do it as well. Oops. Okay, so yes, if we throw the twitches in there, and so let's just keep our, so these will be for the muscle action potential, and then this will be for the twitches. So absolutely, both have 3 phases. All right, make that a little smaller. What else? Excellent. And not both, when we talk about the muscle action potential, both have a rapid depolarization caused by sodium influx. Excellent. And I think different from that, but obviously similar, is both are a big positive signal. Anyone think of anything else?
For the twitch, they both have 1 muscle action potential, which equals 1 twitch.
Sure. Muscle action potential equals 1. There you go. I think those are pretty good. All right, so let's talk about characteristics where they're different.
Their refractory periods, different times.
Excellent. What is the refractory period for a skeletal muscle cell?
About 1 to 5 milliseconds.
Yeah, there you go. Whereas what is the refractory period of our cardiac muscle action potential?
Like 250, 200.
Yeah, it's over 250 milliseconds. Excellent. It's another way they're different.
Their contraction lengths.
So I think that would go more with the twitch. So for the twitch, so we'll do that. The length of the twitch. Excellent. What's the length of contraction for a skeletal muscle cell?
40 to 100.
Yeah. 40, 20 to, it's actually closer to 20 to 100, 17 to 115. Whereas what is the twitch time for a cardiac muscle?
Like over 200.
Yeah, 200 milliseconds. What else? Excellent. I like that. Okay, perfect. Prolonged depolarization in cardiac muscles. So for the muscle action potential, and again, this is why we're kind of awkward going back and forth. So I just want to make sure we, for the muscle action potential, we have the length of the action potential. Notice here, this one is very short, 1 to 3 milliseconds. whereas this one is much longer. We have a prolonged position. What do we call that prolonged depolarization phase?
With plateau.
Plateau, excellent. And of course, we know that it is caused by an influx of calcium, which is another way that these are different. No calcium influx. Here, calcium influx. Excellent. There may be 2 or 3 more, but I think we get the idea. The only thing I would be careful about on a question like this is if I just ask for the action potentials, then don't tell me anything about the twitches. All right, so again, make sure you read the question carefully. How long it contracts for is not a characteristic of the action potential. It's a characteristic of the twitch. So if I ask for similarities in their twitches or similarities in their action potentials, make sure you're giving me the specific pieces of information that I want. Okay. Excellent.
Thank you.
Yep. Any more questions?
Can you go over where the bicep brachii and the tricep brachii are in the arm?
What do you mean by where they are?
Like, I guess they're bellies. I have a hard time finding on the models. It sometimes gets blended in the resources provided by the mastering ENP.
Okay, let's take a look.
More for the biceps brachii than the triceps, but
Give me a second and I'll see if I can find a really good one. Plenty of good bodybuilder pictures, but I want to find a good model.
yeah, I had a hard time finding a good one.
All right, this one's not horrible. I'll start with this one, but let's see if we can find a better one. Hold on, let me keep looking. Because that isn't really what they're trying to show on that one, but there you go. That's a good one. All right. Perfect. All right, let's start with this one first. So, oops. This is the picture that is in your Cosumnes River College virtual anatomy lab. And if you look carefully and closely at this one, like I said, I wouldn't necessarily use this one on the exam for the lateral head, but if you look closely, you can see the dividing structure between the two bellies. Remember, the two bellies are side by side. And what's nice about this is here we can see this one where we see its entire tendon going up to the coracoid process. So this is clearly the medial belly of the bicep brachia. That, of course, makes this one over here the lateral head, but it's not as easily seen on this one here. Here's the next one that I wanted to show you. If you look closely, this one is similar to one of the models we have in the classroom. And again, notice they're showing more the forearm muscles and the deep forearm muscles to be specific. But if you notice, if you look here at the top, this is the bicep brachia. And again, up here on the bicep brachia, you can see the bicep brachia as a whole. But what you notice is there's this nice indentation right there, and I'll remove that, that is helping to show the difference between them. Now, again, not the best picture because it's not showing all of them, but we can see hints of it. The picture that I found in my quick 2-minute search that I think does the best job of showing this is this model that I found right here. Notice, again, we don't see either of their tendons because the deltoid is in place over the top. But what we can clearly see with this model is we can see clearly that there are two bellies side by side. And as you look, you can see that there is a very nice demarcation, an indentation showing you where the two bellies separate from each other. So we can clearly see with this model that there is a differentiation between the two bellies. So clearly now, if I'm pointing, it's way too small, an arrow here and asking you to identify this muscle be specific, and I'm pointing an arrow here, identify this muscle be specific, and let's change it. What muscle be specific is the green arrow pointing at?
The long head of the bicep brachia.
There you go. How did you know?
Because it's on the more lateral side.
Yeah, on the thumb side, it's on the more lateral side. And so then the red arrow be specific would be?
The short head of the biceps brachia.
There you go.
Okay, thank you.
Yeah, so again, if there's that nice demarcation right down the center on the models that does a good job of showing them. Now, obviously, when we look at our bodybuilder pictures, right? Our picture here. Oh, I wonder if this one shows it. Let's see if that shows it. No, I gotta go back now. Notice here, the illustration clearly, again, shows that nice demarcation between the two and shows their insertions very, very clearly.
Are you using illustrations on the exam too?
Some of them, like this one, definitely. I could for some of the deeper muscles, it might be useful, or like, for instance, the internal and external intercostals, or the abdominal girdle, where they were able to separate the muscles independently. Something like that would be good.
Okay.
So notice again here, we can clearly see the medial head very, very nicely, although I guess I should use a different color. The medial head very nicely here. And then again, we can see that nice demarcation that while we don't see all of the lateral head, we can clearly see that they're showing a lateral head. And
Right.
then obviously, what is this one right here? Identify the muscle, be specific. Anyone?
That's the
Medial.
medial.
Yeah, medial
Yeah.
head, absolutely. And in fact, let's go one step further.
It's a short head.
Oh, well, I haven't done anything yet. Said anything yet. Don't answer questions, right? All right. Identify the muscle indicated by the red arrow, be specific.
That long head.
Lateral head. of the
Lateral head?
bicep brachia.
There
The
we go.
tricep brachia.
And lateral head would get your partial credit. What do you need for full credit?
The tricep.
Long.
Head of the bicep brachia.
Lateral head of the,
Tricep brachia.
yeah, this is the lateral head, this right here.
Right.
Yeah.
That's where I'm just confused on like where they're located in the arm for both of them. Like it confuses me. I don't know why.
Because they are anterior, posterior. He's got his arm turned from the side, and because he has his arm turned from the side, we can see posterior muscle,
Oh.
anterior muscle, and the muscle in between.
Okay.
So here, I don't like that red arrow, so let's get rid of the red arrow. So I'll ask the question again. Identify the muscle indicated by the blue arrow, be specific.
So the lateral head of the tricep brachia.
Lateral head of the tricep brachia, excellent. Identify the muscle indicated by the green arrow.
Brachialis.
There you go. Identify the muscle indicated by the purple arrow, be specific.
The medial head of the tricep brachia.
Tricep?
The short of the bicep brachia.
Short head of the bicep brachia. Again, that is the anterior part of his arm. He's just got it turned this way. Right?
Okay.
The view you're seeing right here on the bodybuilder is essentially the exact same view you're seeing right here. Notice the deltoid. So that tells us right now we are at the lateral part of the arm. And notice there is 1 in blue, 2 in red, 3 in green muscles that we can see right here. So identify the red muscle, be specific.
So that's the lateral head of the tricep brachia.
Identify the blue muscle.
Brachialis.
Identify the green muscle, be specific.
The short head of the bicep brachia.
The short, the lateral one?
Along.
Another long.
Long head
Okay.
of the
Okay.
bicep brachia. This view you're seeing right here is the exact same view you're seeing here. The deltoid points right at the brachialis. And so you have 1 muscle on the anterior side of the brachialis, and that is the long head of the bicep brachia. And we know that because the long is lateral. And what else is lateral?
The lateral head
of the tricep brachia. So this view and that view we were just looking at before is the exact same view of the arm.
Where would we see the short head of the bicep brachia on that one? Or is it not possible?
Well, so notice if we instead, I don't remember which direction it was. Here, notice with the arm up like that, identify this muscle, be specific.
That's a short head of the bicep brachia.
Yeah, notice we can see the short head here as well. So yeah, so the short head's going to be towards the inside, it's medial. The long head is lateral.
Can you see the long head and the medial head of the tricep brachia?
On this picture, we can see. So yes, so let's start with this. I would not make you do this on the exam. But notice here is the tendon of the tricep brachia. So notice this kind of going off that way is the lateral head. This here is the long head, and then this here is the mini medial head. So technically you can see it on this, but the same way I won't give you the forearm on this chart, I wouldn't give you this on this. Again, one of the things I love about this class is that this class is hard. And because this class is hard, I don't have to be tricky, right? So I can show you something much more obvious like this. Oops, I guess that's gonna have to do. No, here, let's, really. There we go. Something like this where you can clearly see the tendon. And when you can clearly see the tendon, then you can clearly see those 3 muscles that are coming off of it. The lonely lateral, the mini medial, and the long. Mitch, I didn't miss your question. I saw your question. don't see using the cadaver for any muscle pictures necessarily, I guess, unless it was a really, really obvious one. As I just mentioned, I like to, there's no point in me using tricky pictures because this stuff is already hard, so I don't have to be tricky. It's what I love about this class. However, notice we did use the cadaver to see the fascia lata in the leg. So I think that might be the one obvious place where I might use the cadaver. I don't think, again, this is a test bank I've been putting together for literally 2 years now. So I don't think there are any other cadaver pictures in there, but if there are, then my guess is they would be hopefully super obvious muscles. Kind of like if you think about it on the first exam, the only cadaver model picture I used was for the kidney. Some of you saw, as the organ, you had to identify the kidney inside of a cadaver. And so again, that was something that was super obvious. So I think that would be the other thing as well. So the only time I would use a cadaver would be something super obvious like that. Anything else? Let me remind you again, 3 things. First, again, I have office hours right after this. So as soon as you get off, you think that of something, you can come back to my office hours and ask those questions. I have office hours tomorrow from 11 to 12. I'm always good at responding to emails. So again, even if you don't have any questions right now or haven't wanted to share your questions now, you have the opportunity to get those questions answered. But what I'd also like to really, really, really remind you of on this lab exam, I say this for every exam, but this is especially true on this exam. Read the questions carefully. People often lose points, not because they don't know the information, but because they don't read the questions carefully. Someone gets all excited because they see the arrow pointing here. I know what that is. That is the long head of the bicep brachia. And the problem is I'm asking for the origin, or I'm asking for the insertion, or I'm asking for the 3 actions. So make sure you read the questions carefully. Provide the specific information that I'm asking for. Pay attention to that warning bell. Be specific. If I say be specific, that doesn't mean on this question to tell me that the origin of the short head is the coracoid process of the scapula and the origin of the long head is the supraglenoid tubercle of the scapula. Because while every single piece of that information is correct, you were not specific for the one belly I was pointing at. If I say be specific, that means don't give me everything you know. Give me that precise information about that one belly. And it's either going to be origin or it's going to be name. I remember left and right doesn't matter on this test, but action and part of the body does. Don't just tell me that a muscle flexes. Tell me what it flexes. You have choices. You can give me joint, it flexes the elbow, or you can give me the part of the body it moves, it flexes the forearm. But don't just say flexes. If you just say flexes, you're going to get it wrong. You have to tell me either the joint or the part of the body that it influences. Because as we've learned, there's a whole lot of flexes. You can pretty much flex anything in your body. So give me both the action and the part of the body that it moves. And again, I will tell you how many actions that I want. That is a huge help for you. Because, and again, I haven't done the math, but how many muscles on your list have 4 actions?
A lot.
I wouldn't say a lot. I think for 4 actions.
Oh, for 4?
Yeah, 4
not a
actions,
lot.
I think there might be maybe 5, maybe 6. There aren't a lot that have, let's see. Sartorius, Gracilis, external oblique, internal oblique. Is there any other muscle that has 4 actions?
Did you say trapezius?
Trapezius, there you go.
Okay.
Nope. So 5. So if you see that I ask a question, identify the 4 actions of a muscle, you know it's got to be one of those 5. All right, so again, and if I ask you for 4 actions, I know this is a silly question to ask, but if I ask you for 4 actions, how many actions should you write down?
4.
4. There you go. If I ask for 3, give me 3. If I ask for 2, give me 2. The information on this test isn't hard. There's just a lot of it. That's the trick in this. And keeping all the information organized. That's the real challenge of this. The individual pieces of information aren't hard. It's just a lot of it, and you have to keep it organized in your head. That's the challenge. All right, but I know you guys are capable of doing that. All right, anything else?
Yeah, I had a real quick question.
Yeah, absolutely.
When it comes to the pectoralis minor, the action says protract, depress scapula, or elevate the rib cage.
Right.
It's only one.
Remember, as we talked about, muscles bring their insertion towards their origin. So when that pectoralis minor contracts, it brings the scapula down and it brings the scapula forward. Remember, like we talked about with the sternocleidomastoid and the scalenes, it is possible that I could use other muscles like my trapezius to hold my shoulder in place. So that when I contract this muscle, instead of my shoulder coming down, my ribs go up. So basically I can reverse the origin and the insertion on this muscle so that it goes the opposite direction. And so that's why it's still 3 actions, but it just, notice it can depress and bring it forward, but you can't bring this down and the ribs up at the same time. It's got to be one or the other.
Gotcha. Okay.
So that's basically all I mean by or.
Okay.
Yeah. And you don't even have to put the or. If I point at that muscle and I ask you to identify the 3 actions of it, all you have to say is depress the scapula, protract the scapula, or abduct the scapula, and elevate the ribs. You don't have to say or if you don't want to.
Okay.
Yeah. Great question. All right. I think, any others? Last call. All right, then you guys have an excellent day. Study hard over the next 2 days. Do well on the exam. And then, like I said, take a good 35-minute break. And then I want you diving right into the nervous system. And I will see you guys next Monday. Take care.
Thank you.
Thank you.
Thank you.
Bye. Good luck, guys. I know
Thanks.
you can do this.